Hey, everybody. Thank you for that wonderful talk, Ms. Layton. So I have the honor of introducing our next speaker today. He leads the Michigan State University potato breeding and genetics program and has over 40 years of experience in potato breeding, genetics, and biotechnology. His breeding program has been directed toward the development of improved cultivars for Michigan for 35 years by integrating new technologies like genetic engineering, gene editing, to conventional breeding efforts to target resistance and key processing traits. This breeding and biotech effort has recently shifted to or expanded to include diploid breeding. So Dr. Douches is the co-PI in the North Central Regional Potato Breeding and Genetics Project, and he's also the director of the USAID funded Global Biotech Potato Project for Africa and Southeast Asia, and the lead scientist in developing 4 potato SNP arrays used by potato breeding in the genetics community. Dr. Douches will be speaking today about his work with diploid potato and his program's efforts to develop diploid germplasm that can be used for variety development. With that, I give you Dr. David Douches.
Thank you. So I appreciate the opportunity to come to Texas A&M. Never been here before, and it's a great land-grant university. I'm up there at Michigan State, and my connection to Texas A&M was Creighton Miller. Creighton Miller, colleague as a potato breeder, actually got his PhD at Michigan State University. So there's connections going back and forth. And I learned that Noah, who introduced me, is actually from Michigan. And so today I really want to take the opportunity to kind of tell you something that we've taken on in our potato breeding and genetics program at Michigan State University. I've been in charge of the tetraploid breeding program throughout my career. But just about a decade ago, it kind of occurred to me that there was an opportunity to reinvent the potato as a diploid crop. And so what I'm going to spend this talk today is going through what we've been trying to do to convert potato from a tetraploid to a diploid crop. And so I think I just revised my title a little bit to make it a little cleaner: Diploid Germplasm for Potato Variety Development, Reinventing a Polyploid Crop. And so we always have to have a little background on the potato. It's actually an important world food crop. It's 4th behind the grains. It's the #1 vegetable crop in the world, if I want to be very egotistical. It's also our most popular vegetable in the US. And it's also just highly productive on a per acre basis. Trying to change my slides here. Is there, let's see. Okay. And so the potato has a lot of wild relatives. There's over 100 tuber-bearing species that we potentially could use in our breeding efforts. There's 7 classified cultivated species of potato, and it's a polyploid series from diploid up to hexaploid. Our cultivated potato is tetraploid with 48 chromosomes with a base number of 12. It's vegetatively propagated, so we capture our selection as a vegetative clone and evaluate that through our breeding efforts. There's this term here has been used a lot, narrow genetic base. And so some people will argue that's not the case. Potatoes are highly heterozygous. They're quite diverse. And so we may really not have a narrow genetic base, but we just struggle to make advances. And I think polyploidy is the reason that we struggle to make advances. Well, polyploidy combined with the market needs of the variety. I'm not sure where I'm supposed to point this to get it to change. And so just a little bit of background. So the potato, as in the US, is a $4 billion crop at the farm gate. In Michigan, it's about a $200 million farm gate value. I just like to put up a slide showing the specialized equipment that potato growers need to use. Texas is actually also an important potato state. And so there's a lot of investment in growing a crop. I think this is an underestimate of about $3,000 an acre to get the crop out of the field. And so, and we face a lot of challenges in growing the potato. One, we really need to irrigate the crop to get a productive crop. We're attacked by insects in the north. We have the Colorado potato beetle that is essentially will defoliate the crop if we don't apply insecticides. We have issues with defoliators like late blight, which is a worldwide disease, and the list can go on. Okay, so Fabian, are you changing my slides, or I can't seem to? So this slide is to summarize that the tetraploid breeding is really a long and inefficient process. If you look at the history of potato variety releases, it takes about 15 years from the time a cross is made till the time it's released. We sometimes can accelerate that, but it's really doesn't happen. We're dealing with heterozygous parents, so we're always crossing two heterozygous tetraploid parents. So any trait that we have, any good combinations we have in either parent is lost in meiosis. And so we've never been able, in all my years, be able to fix a trait and move past that and say, okay, we've got that captured. So every time you make a cross, every trait of economic importance is segregated. And so I think that is the real challenge that you have as a potato breeder. And if you were to use wild relatives, which in potatoes we like to do, if you introgress that into the polyploids, polyploidy does a nice job of covering up some of the undesirable traits, but at the same time, if now you want to use that as a parent, those undesirable traits segregate out, so the lines don't seem to be valuable parents because of those undesirable alleles that are accumulating. So we decided a number of years ago, said, let's reinvent the potato as a diploid and create inbreds and move towards F1 hybrids. That's the ultimate goal. And I have a picture here. This is Dr. Shelley Jansky, USDA ARS scientist at the University of Wisconsin. Her and I really felt that this was an avenue that we needed to follow. And so we made an effort to try to move the potato along at the diploid level. A lot of other colleagues in the US are doing the same thing. And I really appreciate that I have the opportunity to talk about what's going on. So this is, plant breeding 101. At diploid level, we can fix beneficial alleles. We can create inbred lines. We can create F1 hybrid varieties. Sounds simple. Okay. So what do we have in our toolbox to get going here? So we have our diploid potatoes. Our diploid potatoes currently are heterozygous, but there's the key to this whole thing is that there's been we can say the discovery of self-compatibility. So tetraploid potatoes are self-compatible, but diploids have a gametophytic self-incompatibility system. And so in my career, it was always said, well, the diploids are self-incompatible, so we can't create any inbreds. Well, if you start going into the literature, you find that people have discovered self-compatibility over the decades, but it was kind of ignored. And there was work then done by Hosaka and Hahnemann in the 90s that discovered a gene on chromosome 12 that contributed to self-compatibility. And they called it SLI, the self-compatibility inhibitor locus. And so with this self-compatibility that's been identified, we said we could introgress this into our germplasm and then move towards inbred potatoes. And so what this also allows us is in our genetics work to actually create new population structures. We always had to cross two heterozygous diploids to create a mapping population. Now we can actually cross or potentially cross inbreds and create real populations down the way, or actually structured F2 populations that haven't been available in the past. So what I want to talk about, oops, somebody, did I do anything? So, what I want to do is talk about these key points today. Is that not all I'm hitting is the pointer. So this is interesting. Okay, so our ultimate goal is F1 hybrids, and so what we want to do is use now breeding strategies that are applicable at the diploid level to actually introgress self-compatibility, and that's going to make our breeding more efficient and effective. And I'm going to talk about dihaploids. So what are dihaploids? It's a term us potato people like to use because the terminology is confusing. We're starting with a tetraploid. We're extracting haploids from tetraploids. So the haploids are diploid. So to not confuse things, we call them dihaploids, but it's probably confusing anyway. So we're extracting dihaploids from our cultivated potatoes to create a diploid cultivated gene pool. Okay, and so we're going to move along, but we also, this working at the diploid level and having some inbreeding possible, it's allowing us to study and access and exploit traits for breeding that we haven't been able to in the past, and I'll talk a little bit about that. And then also one of the advantages of this diploid breeding effort is we work with genetic engineering, but we've also got involved in gene editing. Gene editing just seems so much more feasible at the diploid level than trying to knock out 4 alleles at the tetraploid level. So, and if we have self-compatibility, we can even create a heterozygous knockout and self-it, or select out, Cas9 or something along the way. So the simplicity of having diploids with self-compatibility makes gene editing so much more valuable to our efforts. And so this is just kind of the summary of what we're trying to do. Our first efforts right now have been to introgress self-compatibility using recurrent selection and backcross breeding. And then, plus also doing some R gene selection. Then really, we need to, as we've developed this self-compatibility, start inbreeding the lines, okay? And so that's kind of where we're at, where we've gone through this phase of self-compatibility integration, and we're doing some selfing. And we did some just crosses in the broad sense to look to see if there's any combining ability in an informal way, but we really don't have inbreds to work with or anything at the time. But that's kind of our ultimate goal. So let's just talk about the recurrence selection. So I felt that we needed to have a very aggressive recurrent selection effort to try to introgress self-compatibility. And so we wanted a 1-year cycle just so that we could do this as fast as we can. Maybe that sounds slow to some breeders because what we did was in the greenhouse, we made our crosses in the winter, and then in the winter to spring, harvested those fruit, extracted the seed. Then the seed was planted out into seedling trays, which were then transplanted to the field in June. And then those plants were allowed to grow. We made selections in the fall. And then, well, we made selections based on maturity and tube appearance, tube type, whatever we could see. And then we would bring them into the greenhouse, test them for self-compatibility, and then use those in the next cycle of crossing. Okay. So we felt that self-compatibility was the primary trait, but we also needed to select for a lot of just agronomic traits. And so here's our germplasm we were working with. On the left-hand side, these are what I called our self-compatibility donors. At that time, we really didn't know what the extent of self-compatibility was. So we took a number of donors that were out there. You could see some of these are actually species based. So and then I had actually diploid germplasm that I had been working on for a number of years. And I had created what I considered a germplasm that was made up of 5 different species. And so here it's Solanum chacoense, Solanum freya, Solanum bertholdtii, Solanum tuberosum, which would be our dihaploids, and Solanum microdonum. And they had traits like insect resistance, leaf blight resistance, virus resistance. We were just trying to build, have material that we could cross into our tetraploids at the time. I guess it must be, it's kind of funny how this worked. So, okay, so my student, Maher Al-Thasani, was the one that led this project up. And this work's been published in Euphytica in 2021. I always thought it was Jamie being online last time, that I guess it's just something in-house here. So, and so, okay, yeah, it's like a little game here. Can I talk before the? So as the slides go in and out, if you can get over to this figure here, this is kind of the big point, is that at this time when we started this work, we really did not have any understanding of the genetics other than that we knew that there was this, quote, SLI gene on chromosome 12 in Solanum chacoense, in a selection of Solanum chacoense, which was this first clone we were using. And so we just said, let's go around. And so this early stages of recurrent selection, I wanted to make sure that we did not bottleneck the germplasm too quickly. So we always try to have selections from different populations, half subpopulations that made up the whole recurrent selection population in the first 3 years, so that we did not lose too much diversity. And so then as we got some rounds of recombination and started making progress, after 5 cycles, we got up to 83% self-compatibility in the selections that we made. But while we were doing that, we were also selecting for, tuber appearance, tuber type, and tuber shape, and also considering tuber number. And we were also measuring specific gravity. So when we actually got through all this, we actually had some decent selections after 5 rounds. My one technician would say that in that first, he remembers the first year going out there, and our selection criteria was, did it form tubers? Because we were working with some of these self-compatibility donors that were wild species and unadapted. And after 5 cycles, we had selections that had that were looking like real round white potatoes that may be mistaken for tetraploid. So this was we were quite happy with these results. So the other half of this introgression of self-compatibility starts with the dihaploids, and so in potato we have a pollinator that we can use to create haploids from tetraploids. What this haploid pollinator has is a purple pigmentation. So when we make the cross, we end up getting seeds. And if the seeds have a purple spot on the embryo that we can see through the seed coat, we know it's a hybrid. And if the seeds are spotless, they're potentially haploids of tetraploids, which we now call dihaploids. And so one of the consequences of this process is it's an inbreeding process. So now cultivated potatoes have not been inbred over the years by the potato breeders. So they're full of deleterious alleles, undesirable alleles. And so when we really get to this process, it's a real struggle to get good dihaploids that we can use for breeding. And so I first will say is that this process does somewhat act as like a genetic sieve. And so we get a lot of, well, we don't get a lot of seeds. And then the seeds that we do get, some of them are very poor, don't even make it out of tissue culture after we try to germinate them. And then some come out of tissue culture, make it to the greenhouse, they don't even flower, and they're really weak. And then we do get plants that are that do flower, and then we all know that most of them are male sterile because of the inbreeding process. And then what I've learned along the way is they also have a lot of female sterility. So throwing some numbers up here, we've extracted dihaploids from over 50 tetraploid clones. We try to choose our clones based upon key traits that are important to, the agronomics and processing qualities of the potato. And so so we've been able to obtain about 700 dihaploids from actually 1,750 seeds. So that's like 8 years of work to get that. I think there's worse stories that maybe some of the other potato breeders can say. And I just want to highlight two of my techs, Kate Shaw and Chen Zhang. They were the two that focused on these dihaploid work. And the reason I put them in there was on this project was because their attention to detail. If you're making these pollinations, if you're really sloppy, the tetraploids are self-compatible, so you can end up with a lot of selves, and so you can confuse yourself. And so if you're very careful with your pollinations, yet you end up with only the haploid seeds or the hybrid seeds that have the embryo spot. And so I like to just summarize and say it's a really labor and resource intensive process. And we haven't really found a good way to solve that. And also the extraction of dihaploids vary between clones. Some you get more and some you get less. It's not a matter of effort. It's a matter of just what the clone will give us. I will say also that if you can get enough dihaploids from a clone, it seems like you can find some that are half decent, that they're, and so those are the ones that we've been moving forward with in our breeding program. So now if we have these dihaploids, they're male sterile, and let's say that we know that they're female fertile, we've crossed them with our self-compatibility donor. So if we start with that clone M6, which has the SLI, we create an F1 and then continue the back crosses. So we've been able to get to BC3, BC4. Now, this is a potato back cross. So we do not have inbred lines. So the back cross is really taking just tetraploid dihaploids or tetraploid germplasm, and cross, or I should say, cultivated germplasm into our as our back cross. We're trying to remove the Chacoense wild species and just get it into cultivated germplasm. So it's a germplasm back cross, not an actual back crossing system, because we don't have the inbreds. Just to be clear there. And so we've been able to make good progress in this. And this was less surprising because we knew that we're starting with cultivated germplasm. So we have a lot of good clones that are coming down when you get to that BC2, BC3, BC4 stage, that because they're mostly cultivated, and it's just a matter of what dihaploids have we had at the time as we were going through this process. And while we've been doing this, we've been screening our material with the SNP array to just understand what's going on with our germplasm. And just here's a little dendrogram, and you can see. And so over here is actually a lot of our sample of our selections from the recurrent selection. And up here are actually a lot of our dihaploids and backcross material. So we have kind of two different gene pools. Now, is it going to give us heterosis? I kind of question that at this point. but we do have some distinct gene pools that we're working with. But I wanted to just make a little sidebar, is that when you make a diploid cross, a diploid by diploid in potatoes, you get a lot of diploids, but you also can get tetraploids. And so we also tend to, it's so you can screen them, but sometimes it's hard to tell. Our gold standard for knowing whether they're diploid is to do the SNP array, and the SNP patterns will tell us whether it's a diploid or a tetraploid. And so that's helps us keep the tetraploids out of the diploid breeding program. So in 2021, we ran some field trials to see how our germplasm's doing. And so this was a study, I called it for you guys, the dihaploid hybrid. So there was a dihaploid crossed to something in our diploid breeding program. It could have been some material from somebody else's breeding program, could have been some from our recurrent selection, could have been to another backcross line. But we put them out in the field. We had 2 reps of 10 hill plots. And on this graph, what you can see is where this arrow is. That's kind of I guess a line, say, okay, that's 400 hundredweight is what we would consider an average or yield in Michigan for our potatoes in the industry. And so we're seeing, without any real effort, a number of clones that are having higher yield than our cultivated varieties that in Michigan. And we're also proud that we're harvesting all our diploids with a harvester, not hand picking them up because the tubers are too small. So we've been able to get to that stage. Here's another study where we had, it was a mixture of material from our program, just our best selections. And we ran two reps of 15 hill plots. And here we have the variety Lamoka, and here we have the variety Atlantic. Again, this is about a little over 400 hundredweight per acre, which is kind of a good marketable yield. And then we have all these selections here that are equal or better than Lamoka and Atlantic. So we're quite excited that we're moving forward on this. And then here, just 2 weeks ago, we had our winter potato conference. And so we were talking to the farmers, trying to tell them about diploid breeding. And so what we did was we brought some dihaploids. So here's these little potatoes on the plates. And then over on this side of the table here, we had our diploid selections. I don't know if you can see, but our game was, we asked the farmers to guess which ones are diploids and tetraploids, because all our diploids that we selected had these tuber sizes equivalent to tetraploids. And so this is our demonstration that we're getting the size and the yield along with the self-compatibility at this time, okay? So everything sounds really, really good. However, self-compatibility is still a biological obstacle. It's just not cleanly inherited. We can select for self-compatibility, and in the backcross breeding, we tend to lose it a bit. So it's just isn't as easy to work with at this point. And so this is the challenge that we have to really get to this diploid breeding, is to make the self-compatibility more robust and predictable and durable. Also, because we haven't been inbreeding, there's severe inbreeding depression. In that F1, there's very severe inbreeding. And a lot of times we'll have then male sterility in those F1s, or we get to the F2 and we really hit the wall there. So it's very difficult to get to S3s and S4s in our cultivated material. And as I said, with the dihaploids, we have this issue of male and female sterility. So we got to get over that problem too. And so I mentioned about this dominant SLI gene. And so while we've been doing all this work, the potato geneticist at Wageningen did some work and actually have mapped and cloned the SLI gene. And so we were able to then take the markers for SLI and then look at our material that we have been working with. And so one of my recently graduated students, Dr. Natalie Kaiser, did this in summer or fall of 2020 and published in 2021. And so she went back and looked at our selections in the recurrent selection project and saw that this SLI, that the markers all were increasing as we went through the cycles. And so clearly that we were seeing a lot of self-compatibility, and we were seeing an increase in the frequency of the SLI dominant allele in the population as we move forward. But the interesting thing was that when you got to, started looking at some other clones, there was self-compatibility, but they did not have SLI. So, so in hindsight, we were kind of lucky that we started with more than just SLI in our material. And so our recurrent selection material has maybe more than just SLI self-compatibility. And so we have these clones that are self-compatible without it, which makes sense because the SLI mechanism is kind of unique to potato, and it doesn't fit the other solmonase systems that are out there. So, so that gave us some enlightenment along the way. Natalie also, as part of her PhD thesis, she developed a population that was a cross between M6, which was the SLI donor, and 80-1, which is actually an insect-resistant potato from the same wild species, Solanum chacoense. And she was actually able to create a real population. And so we ended up with 55 self-compatible rills at the F5 stage. And this was published in Crop Science. And so she was able to kind of get some insights on self-compatibility and also on the insect resistance from this work. And so she was able to see that in the inbreeding process that there was a lot of homozygosity, but there was a lot of heterozygosity that was kind of that hung on in the F4 and F5 generations, but it was not consistent along the way there. But it kind of makes sense on chromosome 12 where the SLI locus is that there was some carry along of the heterozygosity. And but we really don't know on chromosome 8 what's happening there. But you can see the rest of the chromosomes tend to be more homozygous. But here was the interesting story is that, so, self-compatibility should be that you put the pollen on the style and stigma, and it's going to grow down the style and be stopped. But what we were seeing is that in Natalie's inbreds that you have you could have a little hard for me to see there. So you have no fruit and no seed, and you see there, you could see that the pollen tubes were stopped. But then on the other three, where you have no fruit, no seed, fruit and no seed, and then fruit and seed, that the pollen tubes grew all the way down. So what is inhibiting, the self-compatibility and incompatibility, or what's the process, is not occurring in the style all the time. It's occurring down there, maybe in the ovule area. So we're really something we don't understand yet at this time. But I'm going to move to now to another student, Will Bailing, who's a current PhD student in my program. And so he's taken on this effort to, we wanted to access some species that were difficult to access because of what we refer to as endosperm balance numbers. And I kind of felt that there was some work in the literature that said, oh, well, if you have self-compatibility, that some of these barriers to species barriers are overcome. So, so he came on board trying to tackle this issue. And so what we've learned over the past couple of years is, so SLI is what we refer to as nail side self-compatibility. And what he's found is that in Solanum verrucosum, another diploid self-compatible species that's from Mexico actually, has what we refer to as a female side self-compatibility. And this is more typical of what we would see in the Solanaceae. And so we see sometimes robust self-compatibility with SLI. We see some interspecific compatibility, but limited interspecific compatibility. But with the verrucosum, we're seeing this robust self-compatibility, which I'll show you some examples of. And then we have the no intraspecific compatibility barriers. And then we have this ability to overcome those interspecific barriers that we kind of see like we see in tomato. But one of the downsides to this is the cytoplasmic male sterility that comes from verrucosum if it's used as a as a female. Okay. So this is one of the things that that Will was able to find out is that SLI did not overcome crosses with species with interspecific reproductive barriers that in the past. So my thought that SLI would help us overcome that was not the case. So here, Solanum pinnatisectum was crossed to 80-1, which is self-incompatible, and it was crossed to M6, and it's hard for me to see that from there. Okay, so 80-1. Okay, you see that it grew down the style, and also in 80-1 for pinnatisectum, but for bulbocastanum, it didn't overcome those barriers. So it didn't matter whether it had SLI or not. Okay, whereas Will took Solanum verrucosum, and he was able to take these and other species and have the pollen tubes go all the way down and obtain hybrids between verrucosum and these species that we have been has been difficult to introgress. And what he argues is that this self-compatibility that we're seeing, this female side self-compatibility, is more predictable and robust. Here's just 4 progeny. So it was segregating in a very simple Mendelian fashion. But if you really look at the argument he was making to me was that the 3 that are self-compatible are broadly self-compatible. You're getting good fruit set, good seed set, and then clearly no seed fruit set there. And so that the verrucosum-based self-compatibility may be a better system to use than the SLI system that was initially described. And so he's also just fanatical about species, and so he's been looking for other species that have self-compatibility. He's just screening various ones in the greenhouse, and so we have this little short list here. We've got verrucosum, which we're talking about. Natalie found some self-compatibility in Chacoense that was non-SLI, as we saw with the SLI markers. Will found that in Curtianum and Polyadenium. One's a South American species, the other is a Central American species. And Will's found it now in pinnatisectum, another Mexican species, and also in Solanum indigenum diploids. So, and I think if you go in literature, you'll find it. So we're really getting excited that there's more to self-compatibility than SLI. And we may have kind of got onto a new track that the verrucosum-based self-compatibility in the long run may be the better way to go. I want to kind of switch gears for a couple of minutes here and come back to Natalie. And so this was, I felt, was with one of the opportunities that diploid self-compatibility gave us was to actually maybe tackle some traits that have been difficult to work on for decades in potato breeding. So there was an effort in the 80s they identified Colorado potato beetle resistance based upon a glycoalkaloid called foliar glycoalkaloid called leptines. And so then it's been worked by the breeders, and when you cross it into tetraploids, it just dilutes out and you end up with nothing. And so I thought, and there's also in literature, there's some cases that says that some of the alleles may actually be recessive, or the genes may be recessive to give us the trait that we want. So if we can work it at the diploid level with self-compatibility, we might be able to fix some of these recessive traits and get the leptine-based resistance that we need. And so it's just leptine is just a glycoalkaloid that's built off the solanine and chaconine. Not going to go into that, but it works well against the beetles. So this population that I mentioned earlier, this is actually what the plants look like. It's an intraspecific cross. We thought this was a better way to study it rather than making an interspecific cross and complicating the genomes there. And we had enough variation in the SNP array to get good coverage. And so Natalie was able to create those inbreds, take them to the field, and actually select a large number of F5s that had good levels of insect resistance. And she was able to map the resistance to a QTL on chromosome 2. And right now we feel that there's a candidate gene that we're going after. And our goal is to see if we can use CRISPR to knock that candidate gene out and kind of confirm that that might be a key gene in the leptine pathway. And so now our efforts are trying to cross this resistance into our diploid germplasm. And so we're making the effort to create those populations, take them to the field and make selections. But in the meantime, coming back to Will, he's working with the varicosum. And so he was able to cross varicosum with a species called Jamesii, which is actually a species that is also in North America here. And this has insect resistance. So Will actually crossed varicosum with Jamesii, which had barriers prior. And then he crossed that hybrid to one of our diploid breeding lines. And so this clone is 50% cultivated potato. And it might be a little hard to see, but I put a circle there. So what was very interesting about this resistance is that the beetles came to the plant, actually ate, and then they immediately died. It was really satisfying to see something like that as a human, whereas Natalie's leptine-based resistance, they come to the plant, they'll eat the plant, and if they don't eat enough to die, and they kind of walk off and go do something else, and try to find something else, and they'll come back if that's all there is. But with Will's, it was kind of like, hey, they came and ate, and then he died. And so it was really, really exciting to have something like that. So Will's materials kind of his approach is allowing us to access some germplasm that when Natalie started, she tried to use but couldn't use because we hadn't overcome those interspecific reproductive barriers. So this working with this bridge species Solanum varicosum is kind of having us kind of reevaluate this whole concept of the reproductive barriers. So it comes down to, you got pre-zygotic barriers and then you got post-zygotic barriers. And so we're trying to clarify that. And so the work with SLI and with varicosum is taking us in that direction. And so for people that are in the potato world, they've seen the term EBN1, and that refers to endosperm balance number. And it's a system that's been developed to help us understand how to make crosses at different ploidy levels. Don't want to really go into that, but I'd rather say that the EBN1 species have interspecific reproductive barriers that we can overcome using Solanum verrucosum. So this EVN concept is kind of falling apart for us from a breeding perspective. We're able to access this material at the diploid level that we haven't been able to in the past. And so there's lots of traits in this material that's from Central America and Mexico. And so we're just wanting to grab that. So like in the past, the only way we could do this was to take these species from Mexico or Central America and do like somatic fusion hybrids, create like hexaploids, and then back cross them to tetraploids, create pentaploids. What a mess. And then you end up with material that has the genes in there, but never are good enough to be varieties that the farmers can grow. I'm kind of bullish that maybe we can get there by diploid breeding, because we can then back cross out a lot of the undesirable traits like they have in the tomato world. And so I'm really seeing this verrucosum as a bridge species that puts us on par with tomato in introgressing some of these wild species. And here's Will's taken some other species, and again, this is like 50% cultivated potato, and we're getting, you know, what I consider acceptable tuber types with acceptable maturity in just a couple crosses. So this is just, you know, starting things out. And so just the last step I was mentioning about the gene editing. And so we've been involved with gene editing, and what's nice about gene editing versus genetic engineering is the way the regulatory system is set up, that we can actually maybe grow gene edited plants without regulation in the US under certain circumstances. And so my student, Dr. Felix Enciso, and a postdoc, Swathi Nadadakudi, they worked on self-compatibility through gene editing. And so they knocked out the sRNase locus and achieved self-compatibility. This was published in 2019. And this has been our gene edited version of self-compatibility. And it's what got us going along this lines is in tomato, we know it's a dysfunctional sRNase that gives us self-compatibility. And so we believe we know that we feel that this verrucosum self-compatibility is sRNase based, and that there's a lack of sRNase function in the style. So this gene edited version is an alternative to that. In addition, there's other genes that can contribute to self-compatibility, incompatibility. And one is this HTB. Well, there's HTA, HTB. We really wanted to knock out both. We weren't able to knock out A, but we knocked out B. And so here's our sRNase knockout line, and here is our HTB plus sRNase knockout line. So what we learned is that when we knocked out both of those genes, that we got more seed set in our fruit than just the sRNase by itself. We want to follow this up and knock out HTA along with HTB and see where that gets us. But right now, we're seeing an improvement in seed set by knocking out HTA along with the sRNA. And then the last two things to mention is that my student, Talani Jayakoti, a PhD student, one of her efforts is to create diploid germplasm that we can use in the lab for gene editing or genetic engineering or studies. You know, so she just published in the American Potato Journal on this, and so she tested a number of lines for their self-compatibility, their tuber production, their ability to regenerate in transformation. You know, so we feel like we've been able to identify some lines that can be used in research that have some agronomic value rather than just be a complete lab rat. And lastly, she's also, one of her aspects of her PhD, she's been sequencing one of a doubled monoploid line that we have. So the original doubled monoploid that gave us our potato genome was sequenced a number of years ago. It's not a good clone, but this clone here, developed by Richard Bayou at Virginia Tech, is a one that we can use better in our breeding and genetic studies. And so she's done the sequencing. She's almost done assembling it. So hopefully we'll get a publication out in 2022 on this. But it gives us some more genetic resources to work with. And the verrucosum line that's giving us the self-compatibility and bridging is also being sequenced. So we're trying to develop those resources that we can do at the diploid level that we don't see in, or make that's difficult at the tetraploid level. So summarizing everything here is, I feel that 2x breeding is more efficient and effective. It allows us to potentially to fix or introduce traits that we haven't been able to in the past. So we really haven't gotten to that point of fixing the traits, but we are introducing them into our germplasm at this time. Self-compatibility, has to be studied and understood and utilized. And so there's a lot of work that still needs to be done for us to grab a hold of everything, but we're feeling bullish about this female-based self-compatibility coming from verrucosum, not to poo-poo the SLI-based, but this is, we think is better in the long run. And working at the diploid level with the ability to inbreed allows us to actually maybe study traits better, and which will allow us to use them better in breeding. And we tap the wild species so often, it's necessary that we have this diploid breeding system. And lastly, the diploids allow us to access these genomic tools, the gene editing and the sequencing, so much more easier than at the tetraploid level. And so just the last slide here is just to say that we have a USDA grant. Jeff Endelman at Wisconsin is the lead on it. And then I'm a co-PI, Robin Buell at University of Georgia, and Laura Shannon at Minnesota are key people on this project. And so we're trying to tackle these issues collectively as the public breeders here in the US to improve the self-compatibility and fertility, to broaden the genetic base by extracting more dihaploids rather than just at MSU, and hopefully develop some key inbreds for different market classes that come out of this. And in the long run, maybe lead to actually a seed-based F1 hybrid system rather than a clonal potato release. So that's my story today. This is my team in the lab. I have a great team of students and staff. And so at this time, I'll take some questions. I ran a little over, but thank you.
Thank you for that great talk, Dr. Douches. At this time, we're going to open it up for questions. So if you have a question in the Zoom chat, please raise your hand or put it in the chat. If you have a question in the room here, please raise your hand and one of our mic runners will come to you. So.
Hi, Dr. Douches. Thank you for the impressive presentation. So you are telling that the diploid breeding program is taking over the tetraploid breeding. So what is your perspective on the industry prospect? And then what do you think, like the industry will take over the breeding program, or the public sector will continue? Because now the public sector is more leading than the industry. What is your perspective on this? Thank you.
Well, so I think that we're still doing our tetraploid breeding, but the diploid breeding's taking up more of we're investing more resources from our pool that we have. And if I still bullish that diploid breeding will get us there. And so I feel that we're answering the question about yield and size. Now we're focusing on the market traits. And so it's going to take a number of years to get those market traits in order. But I still feel the efficiency of the breeding at the diploid level and the ability to create, do some selfing. And actually the traits seem to segregate out more simply at the diploid level, that we're going to make progress faster. But if this whole thing blows up, the diploid germplasm can still be introgressed into the cultivated. And I think it still will be useful, but I still think in the long run, the diploids will take over. And what I was explaining to our potato farmers in Michigan is you're just going to get a potato. It doesn't matter whether it has 24 chromosomes or 48. If it does what you want, that's all that matters. So they don't really need to care. They just need to support the breeding efforts in the US. I think.
Okay, David, one question. I mean, there's no doubt that breeding on a diploid level is going to be a little more efficient than tetraploid level. But what might you be losing by going to the diploid from a tetraploid? What traits or qualities of a tetraploid you're not seeing in the diploid?
Yeah, that's a good question. So I don't have a lot of things that the polyploid favors, other than right now our tetraploid, germplasm has more consistently high yielding. So that's there. But the ability to advance it is where we lose steam. So we seem to be stuck in this kind of okay high yield that the farmers accept, but we're not able to jump anywhere. And I kind of feel that as we move forward with the diploid breeding, we could actually create recurrent selection populations that will improve for yield that we haven't been able to do in potatoes. So I really don't have a lot of tetraploid features that I'm losing. It looks like Isabel there.
Question, Center.
Hi, Dave. Great presentation. Thank you. I was wondering if in the process of developing the dihaploids and then moving towards inbred lines, you have uncovered interesting traits that were not present in the original tetraploid mothers. So that would be traits that are due to recessive alleles that then you uncover, and it could be based on morphological aspects, agronomic aspects, or even disease resistance, maybe some disease resistances that you were not even aware of, but now because you have recessive alleles that you uncover those.
Yeah, so I don't know if we've, that we've achieved the full potential of the inbreed or anything yet, but one of the things that we're seeing out there that we couldn't explain this summer is that there's some virus resistance that we're seeing in the field that is not based upon the R genes that we've been using in our tetraploid breeding program. And it's not the Chacoense even, though it came from the diploid. We're not sure where that's coming from, but it's clear that we've had material in the field that's not getting infected, very surprisingly. So that's the one thing. But I was really impressed, and I couldn't show all the data, but I said that we made improvement for all these traits. So we were basically making visual selection in the field for tuber appearance and size and bringing those back in and then selecting for self-compatibility. But we were seeing this improvement in specific gravity, and also scab resistance without even trying. So there's a lot of traits in there that we weren't even really selecting for yet, but clearly that it's there. So all we have to do is now apply a little more selection.
Yeah, in spite of the difficulty you mentioned on the breeding for tetraploid, there has been the progress in potato breeding is very impressive, and the yield levels are very high. So I hope that this diploid thing will catch up with that someday. But you didn't show any quality differences between the two, starch content, protein, and so on. Would there be any drawback or something that if you go for diploid breeding? Thank you.
Okay, yeah, Great question. So we purposely haven't selected for processing traits yet in the material. Actually, when I was flying here yesterday, my crew was doing some storage chip processing tests yesterday on some of the material that you saw some data on. So, but I'm not expecting to see a lot at this time. But from a protein perspective, we are seeing, we did screen our material for protein content. And so very interestingly, we screened our advanced tetraploid material. And then we took some of the lines from that hybrid diploid study. And it was almost like you put the two graphs together, and where the high protein and the tetraploids left off, the diploids started and went up. And so we're real excited that in the diploid germplasm, we're seeing a higher protein content. And also my student Will is working with one of those Mexican species that is noted in the literature of actually having an extremely higher protein content. And so want to get that tested soon to see where that falls in on our material that we're screening. But yeah, we're seeing an advantage in the protein. Got a question coming from the chat at the back there.
Hello, my name is Denise Greer, and I have a question. I'm not sure if you can answer it, but when you were talking about the extra protein content and you're talking about just the fact that there's a $4 billion production with regard to potatoes, what type of niche markets are included in that, where this where the dihaploid and is important? What type of niche markets? Because I know, we always talk about food security, but what type of niche markets, if you understand the question?
Well, the market right now, the diploids are not on the market of any real commercial level. This is really going on in the breeding programs at this time. But the $4 billion is a farm gate value. Then it goes into frozen processing, into chip processing, fresh market, and seed. But then there's also within there's the starch market, and to go along with the protein, the Europeans have been extracting protein from potatoes because the potato protein is actually a well-balanced protein, and it's one of the better non-dairy proteins that can be used. But the current level in cultivated potato doesn't make it economically feasible to just grow potatoes for protein and extract it out. And so our long-term goal is to maybe see how high can we get the protein, and can it get to the point and to be a protein extraction process that, grow the potatoes for protein. Like they grow potatoes in Europe for starch, and so they grow these big ugly potatoes just for starch. We might be able to produce some big ugly potatoes just for protein, which would be really nice.
So not yet viable in the market from that from the perspective of extraction for things outside of farm or food. Is that correct?
Yeah.
But
not on any major level. No.
Okay, thank you.
Just back to your slide on the beautiful effects of the leptinine on the potato beetles.
Mm-hmm.
Can you speak a little further as far as the effects of that chemical, that glycoalkaloid, on human toxicity in food trials?
Good question. So what's good about leptines is I don't know that I'm assuming that its toxicity on humans would be similar. However, what's special about this leptine glycoalkaloid is it's foliar based. It doesn't move into the tubers. So that's why it's been a valuable host plant resistance to utilize by the breeders.
I think we got time for one more, Dr. Balensberg.
You mentioned several traits that you were looking at that I would call food quality traits, but the one you didn't mention that seems most important to me is the low calorie potato, or the one that doesn't absorb as much grease when they fry it. Do you have any work that looks at how to actually reduce calorie content in the potato or make it less digestible?
Well, the potato doesn't have any fat. So the fat, all the fat that's added is in the process. And so I think actually, if you looked over time, that in the French fry and potato chip industry, that there's been an increase in potatoes for starch content. And if the more starch content you have, the less oil absorption there is. And so I feel that especially in the chip industry, we have now higher solid potatoes. And so actually your chips are not as greasy. So I can't give you the figures, but I know that clearly, so the processors benefit because they use less oil, which saves them money, and they actually get more yield per pound of potatoes. So they really like that high solids trait.
All right, I think we'll wrap it up there. Thank you again, Dr.
All right,
Douches.
thank
That's
you.
great.
Welcome back, everyone. Our next four speakers are Texas A&M graduate and undergraduate students who were selected out of many applicants to present their research. The first speaker is Daniel Crozer. He is a PhD student in the sorghum breeding and genetics program under Dr. William Roney. He came to the Texas A&M after completing his undergraduate in plant biology from North Carolina State University. His research focuses on breeding for herbicide tolerance in sorghum, evaluation of elite temperate adapted germplasm developed from tropical sorghum lines, and predicting hybrid performance from the inbreed genotypic and phenotypic data. He aims to continue furthering to help meet the growing future needs of the world.
So thank you, Oakson, for that introduction. So today I'm going to be talking about how we can use inbred phenotypic data and hybrid grain sorghum prediction models, and then also the relative importance of modeling dominance and G/E effects. And so I'm really glad we already had a speaker come and talk about hybrid sunflower production because it makes this slide much easier. Sorghum's commercially grown is a hybrid grain crop, and we utilize the same CMS system or a similar one that they would use in sunflower. And so the breeding pipeline, we initially focus on line development, and then at some point, say the F5 generation, we switch to hybrid evaluation. And so you can see in the schematic here, a rough diagram of how a breeding program works and the effects of selection within there. And genomic prediction is a tool that we can use within breeding programs to increase genetic gain. And how we can do this is, if we can account for the variance covariance between related individuals, we can better predict observed individuals and also unobserved individuals. And so the way we account for this relationship between lines, or one way we can is by augmenting the mixed model equations. You may have seen this before where you have your fixed and your random effects modeled in like a matrix-like format. And then, so for the GBLUP approach, you include this G matrix, which is the relationship between lines. And so genomic prediction has several roles in which you can implement it in a program. I've put some here on the side of these figure in this here, genomic informed breeding decisions. I'm going to focus on two, specifically predicting hybrid performance and specific hybrid combinations. And so the objectives of my study are to assess the relative importance in modeling dominance and G/E and hybrid grain sorghum, and also determine if inbred parental data can help predict hybrid performance. And so the population we used to study this had 92 inbred parental lines. There were 46 AB lines that were seed parents, and then 46 R lines that were pollinator parents. And we crossed these in a partial factorial to produce 395 hybrids. And we grew these in 4 environments in a replicated trial. And so these are grown in 2019 and 2020. If you'll notice, both years in College Station, we grew all 395 hybrids. But then for our South Texas locations, which would be Taft and Lyford, we only grew about half the hybrids, just because these are large trials, so they're difficult to manage for space sometimes. And in addition, in 2020, we grew the parental lines in a separate but adjacent trial alongside the hybrids in both locations. And so you can see a distribution of the blues from within each environment in this figure here. And so you can see there's significant genetic variation within environments as well as between environments. And you can also see the effects of heterosis within an environment where you compare the inbreds, which are the red bars, to the hybrids, which are the blue bars. And so you can see our hybrids are higher yielding and generally taller than the parental lines. And so the genomic prediction models we used were, first we have to talk about the genetic data. So we had roughly 38,000 SNPs from GBS sequencing. And then we built prediction models from these using kernel-based G. There's something weird going on in this upper right-hand corner, this checkerboard effect. These are where the inbred lines are. And so there, you can see the population structure present between our heterotic groups where we have our A, B lines and their R lines distinctly separated. And then you'll also notice correspondingly where those inbred lines are present in the dominance relationship matrix. And because they're inbred, their expected heterozygosity is really low. And so we ran a series of Bayesian regression models to predict the performance of material. And we used a cross-validation scheme where we had 50 replications, where the data was partitioned in a roughly 70% training, 30% testing. And we used two cross-validation schemes, one where we removed all hybrids for a given line. That would be, you have no test cross information, no hybrid data for that line. That's CV1. And then CV2 is where we removed some hybrid data, but not all hybrid data for a given line. So that would be where you may have a line crossed to one or two other lines, but you don't, and then you're trying to predict a third or fourth or fifth hybrid combination. And so these are results of the prediction models. These are the hybrid only models. So there's no inbred data here. And so you can see here, these are the prediction accuracies, and it's split up into 4 quadrants, where the height of the bar is the correlation coefficient. And then if it's hard to see differences, I put letters to indicate statistical differences between the bars. So if you look right down the middle, you'll notice CV1 is on the left and CV2 is on the right. You'll see the prediction accuracies were much higher in CV2. This is because CV2 is easier scheme to predict in because we have more information. We have, hybrid data to help us predict other hybrid combinations. CV1 is a much harder scheme to predict in. And then if we also look at the differences between the two traits, plant height and grain yield, plant height is a much more heritable trait, so we have much higher prediction accuracies for that. And then if we look within these at the bars to compare the different models, so if we look at an additive effect only model for grain yield is compared to an additive by environment effect model for grain yield, you can see there's a slight increase in prediction accuracy. However, when you include dominance effects, so that one to that one, you don't really see the same increase in prediction accuracy. And I think one reason this is because if you look at the variance decomposition for these traits, specifically this one, you'll see we are partitioning variance into dominance effects. However, we're mostly partitioning that variance out of the additive genetic effect term and not out of the residual effect term. And then I think it's also a limitation of the G-BLUT model framework where you're modeling the relationship between individuals is why we're not seeing dominance play a larger role. And then so the next part is, how do we include inbred phenotypic data and why would we? Well, typically parent progeny are correlated in terms of performance. However, heterosis can mask this relationship. So I just did a correlation to look at how correlated are parents to their hybrids for these two traits. So mid-parent is the average of the two parents. And so you can see for both these traits, we do have significant correlations. So taller inbreds tend to produce taller hybrids, and then same for grain yield. And then so that leads us to and how well does this information improve our prediction models? And so here I've simplified it. I've just showed one model. So there's no different model types. There's one without inbred data, so hybrid only data, and one with inbred and hybrid data. And you can see that for the CV1 scheme, which is the harder scheme, that's where you have no hybrid information for a given line. we do see a pretty significant increase in prediction accuracy. It's about 36% for plant height and 15% increase for grain yield by modeling the inbred parental phenotypic and molecular data to account for some of that missing data. But you'll notice the CV1 scheme, even with inbred data, is still lower than that of CV2. And you'll also notice in CV2, we don't see any statistical difference between the prediction accuracy of these two models. So what that tells us is inbred phenotypic data is not as good as having actual hybrid data for predicting hybrids. But if you include inbred phenotypic data, it can make up for some missing data in your models. Okay, here we go. And so to kind of visualize genomic prediction and see how and why we can use it, what I've done is I've plotted for the CV2 scheme, which is the easier scheme for grain yield, the predicted versus the observed values from a model. And then I've imposed a selection criteria which says, if we were to drop the bottom 50% and keep the top 50% based on what we predicted, how good is that in terms of actual progress? And you can see the means up here for those two. And so we've shifted the population mean by about 500 kilograms per hectare for grain yield by using a selection cutoff of 50% for grain yield. And so that's pretty significant. And that's how genomic prediction works. It just advances the population. But you will notice the highest actual performing hybrid is way up there. And it's just predicted as just below average. So we would have not selected or advanced that. And I mean, you don't want to see that, but it's also kind of a reality you face with even phenotypic selection for a lowly heritable trait. You have that risk of not selecting one of your most elite lines. And so that brings us to the conclusions, which are modeling G by E improved prediction accuracy in this population while dominance did not. And then modeling inbred parental data can improve the ability to predict hybrid performance in the absence of hybrid phenotypic records, such as that of CV1 that I presented. And so with that, I'd like to thank everyone that helped me do this research, my advisor, Dr. Rooney, as well as the whole sorghum lab for helping make the crosses, phenotype this stuff, develop the models. Dr. Patricia Klein and Dr. Bob Klein for their molecular work, providing high quality molecular data and expertise of how to treat and handle that. And then the rest of the faculty and staff here at A&M. And then also the committee and the sponsors of the event for letting me talk today. And then I'd also like to thank the Sorghum Checkoff program and then the Borlaug Monsanto Chair for International Crop Improvement for some financial support of this work. And with that, I'll take any questions.
On the podium. Great. Thank you, Daniel. So we got time for just a couple of questions here. So again, if you have one in the chat, please raise your hand. Otherwise, raise your hand in the crowd. Got one over here to the left.
Okay, thank you. It's a good job. Maybe I'm somewhere missed. You predicted from parents direct predicted hybrid, or you use hybrid predict hybrid?
So the two prediction schemes where we had hybrid data and we left some of that out, using only hybrid data from related individuals predicted hybrids. And then the other scheme, and then you could also include inbred parental data in that, where you still have hybrid data, but you now have inbred parental data as well. Incorporate that into your prediction.
Because if you can genotype the parents and use the parents genotype directly predict hybrid, you don't need to make cross. Certainly, when you're training the model, maybe you need a limited number of hybrid make. And that's why I ask, because there are quite a few publication people use genotype parents directly predict hybrid yield, plant height, whatever the trait.
So that's what we did. We sequenced the parents, and then the hybrid genotypes were traded in silico, and we already have the parental genotypes from direct sequencing them.
Okay.
So we did sequence the parents, and then we created the hybrid genotypes in silico. So we do sequence the parents, but you still need to collect data on hybrids in order to develop those prediction models.
Yeah, when you're training the model.
Oh yeah.
Yeah. After training that you don't need it, I believe.
Thank you.
Talk about one more. All right, great. Thank you, Daniel.
Thank you.
All right. Thank you, Daniel. Our next speaker is Jasmine Yu. She is a PhD candidate in the Department of Biochemistry and Biophysics at Texas A&M. She studies the physiological and molecular mechanism of energy sorghum stem development. Her research focusing on understanding hormone signals pathway during stem growth under shade condition. She believes her findings will contribute to improving lignocellulosic biomass yield from energy sorghum and making it a better biofuel crop. Outside of research, she is interested in science policy, K-12 outreach, and taking care of her 2 rabbits.
Hello, so today I'm going to be sharing our data on stem intercalary meristem development and transcriptome dynamics in bioenergy sorghum. So the world population is projected to surpass 9 billion by the year 2050. And with this increase in population, we're going to expect an increase in demand for energy. However, currently, a vast majority of that energy is being derived from fossil fuels such as coal, natural gas, and petroleum. Not only are these sources of energy limited, but they also have long-standing negative impacts on the environment and human health. On the other hand, biofuels are a potential alternative carbon neutral fuel source. In 2020, around 12% of the total energy consumed in the United States was derived from renewable energies, and a large portion of that came from lignocellulosic biomass, which is derived from plant cell wall materials. Now, qualities that make a good dedicated biomass crop include high biomass yield, low inputs, growth on marginal land, as well as resilience to drought and pests. Oh, cannot hear? Oh. So our lab studies bioenergy sorghum because it fits all of these qualities. And my research is specifically interested in sorghum stem regulation and growth because around 84% of total sorghum biomass is accumulated in the stem. So prior research has focused on stem regulation. So, one of these has been understanding and characterizing the dwarfing loci in sorghum, which controls internode length. So DW1 has been found to be involved in brassinosteroid signaling, DW2 lignin lipid signaling, and DW3 auxin transport. And today we're going to share research on increasing our understanding of when and how stem growth and composition is regulated using morphological analyses as well as transcriptome analyses. Because we find that the stem is made up of many different sections, and each of these plays a very special role in how the plant develops as well as accumulates biomass. So when we're looking at stem morphology, we're really focusing in the vegetative stage. And during the vegetative stage, a phytomer is produced at the shoot apical meristem located at the top of the sorghum plant every 3 to 4 days. And a phytomer is comprised of a leaf blade, leaf sheath, and a phytomer stem section. Now focusing in on the stem section, the phytomer stem at the top is the nodal plexus, and this is where the sorghum leaf sheath enters the stem. And then below that is the internode, and below that is what we call the pulvinous tissue, which is usually studied for gravitropism responses, as well as the presence of tiller buds and nodal root bud formation. And if we focus in on the internode, there are different zones of development, starting at maturation at the top, elongation and division, and the size of these zones really depend on the stage of growth. Today, we're only really going to focus on the zone of division or the intercalary meristem, because based on the literature, it's not very well known exactly where the intercalary meristem is located and where it begins. When we look at the morphology, we see over time, there's an appearance of a white band of tissue in this intercalary meristem zone, and appears around when the internode begins to elongate around phytomer 6. And then over time, it actually turns into a brown color until around a very mature phytomer, it turns into an indentation, seeming to separate the internode and the nodal plexus. So while we're looking at this, we're really wondering, what exactly is this white band? Is it significant, and is it different from all the other tissues in the stem? So to understand this, we looked at cell morphology data, specifically as cell parenchyma cells across different sections of the stem. So we notice here at the longitudinal cross sections that the nodal plexus and pulvinus are very similar. They have very disorganized cells and very different shapes, especially compared to the mid-internode, where the cells are very uniform in shape and have this vertical organization. And then when we look at the white band, it seems like the top of it is very similar to the mid-internode and the bottom of it is similar to the pulvinus. This tells us that the white band is very likely this transition layer between the internode and the pulvinus, and very likely even a boundary layer based on our transcriptome analysis data. In addition to looking at cell morphology, we also looked at MRI images, which allowed us to create a 3D image of a full part of the sorghum plant we were interested in, and then look at cross sections of specific parts. So starting here at the pulvinus at the top, you see these white circles or half circles, which are signaling the location of vascular bundles. And it's very uniform throughout the stem. And then around it is the leaf sheath. Focusing on the right side, you see there are white lines indicating that the vascular bundles from the leaf sheath are entering the stem at that point. They're entering at an angle. This is even more clearly seen in the nodal plexus, where the leaf sheath is entering the stem completely at that point. So there's no leaf sheath around the nodal plexus. And when we look at the white band tissue, you notice how the vascular bundles are very much smaller compared to any of the other tissues. And altogether, this really just tells us how different the morphology is across the stem. And focusing back into one of our main questions is of what exactly is the white band and where is it localized? We did a growth analysis experiment where we exposed a growing phytomer and placed colored dots to represent different parts of the stem. So purple for nodal plexus, blue for pulvinus, and red for mid-internode. We covered it back up and then let it grow for 4 days, and then looked at how these dots were displaced, and found that the nodal plexus dot and the pulvinus dot didn't change at all, but there was a very large displacement between the mid-internode and the pulvinus. And this tells us that the cell proliferation is in fact occurring at the base of these growing internodes and not much at all at the pulvinus or this nodal plexus region. And to get a better understanding of the localization, we did some transcriptome analysis focusing on the growing zone of the stem, phytomers 1 to 5, we call the apical dome, phytomer 5, and then growing tissues from 6, 7, and 8. And from 6, 7, and 8, we collected tissues specifically from the nodal plexus, the top of the internode, the bottom of the internode, the white band, and the pulvinus. And we looked at the number of genes that are differentially expressed across these tissues and find that the nodal plexus and pulvinus, as well as the base of the internode and the white band, have very low amounts of differentially expressed genes. And this indicates that their stage of development and transcriptomes might be quite similar. And because we're so focused on the intercalary meristem, we're particularly wanted to see what was happening between the white band and pulvinus, because although they're so close to each other, they are different morphologically, as we saw from the cell morphology data, and they also have a very large amount of genes that are differentially expressed. So we looked at gene ontology analysis data to see the types of gene functions that are enriched in the white band and pulvinus. And what we see is that in the white band, cell proliferation type of genes are highly enriched, such as cell division and cell cycle, and compared to the pulvinus, which is highly enriched in secondary cell wall formation genes, such as lignin biogenesis. And this tells us that the white band is likely spanning the intercalary meristem region. So for a more higher resolution analysis of our transcriptome, we're pulling out the gene expression data, or the TPM values, of certain cell division and cell wall biosynthesis genes, and showing the expression across the stem tissues we collected. On the left being the young tissues at the top, and on the bottom, on the right at the bottom of the plant, where phytomer 13 is. And with the cell division genes, we see that it's highly expressed where it's expected at these very young tissues, as well as the intercalary meristem tissues, the base of the internode and the white band, and also decreases over time as the stem matures. And then when we look at the cell wall biosynthesis genes, they are highly expressed in the opposite types of tissues, and they're maintained over time in the nodal plexus and pulvinus, and they also increase in the intercalary meristem. So this information, again, just further validates that the stem intercalary meristem is in fact located at the base of the internode, including the white band, but above the pulvinus. So additional studies we'd like to go into include understanding how the cells are transitioning from division to elongation and maturation in the stem, as well as considering factors that are modulating expression, such as miRNAs and signaling peptides. And in conclusion, we really hope that the information we've collected from morphology, as well as the transcriptome, can produce testable hypotheses and provide a baseline for comparison of other experiments done on bioenergy sorghum. I'd just like to conclude by acknowledging committee members, advisor Dr. John Mullet, collaborators, and especially Dr. Joel Oliver, who recently graduated and played a significant role in this project with development, experiments, and data analysis. Oh, and I am ready for questions.
Thank you, Jasmine, for that great talk. So we got time for a couple of questions. If you got a question, just raise your hand. Our mic runners will come to you. Questions in the chat, if we have any. Anybody? All right, right there.
From the microscopic analysis.
Please use the mic so the people in chat and everybody can hear.
I have a question about how do you distinguish the pulvinus and the nodal plexus? Because from the microscopic analysis, they're pretty similar, and there are upper nodal plexus, and there should be another nodal plexus below the pulvinus. Why, when you plot your image, why you didn't define the lower nodal plexus to be with that internode?
Okay, that's a really good question. That's actually another part of the project, and there are different theories of how the stem is growing, whether or not it's separating the nodal plexus or if it's just growing out from the white band. But to answer your question about the microscopy data, the way we did it is we took the longitudinal cross sections and cut it into specific sections and then kept track of it to make sure that the pulvinus was not the nodal plexus.
Any further questions? All right, if that's the case, then we'll wrap up. Thank you again, Jasmine.
Howdy, everyone. Next up, as our last graduate student Texas A&M speaker is Brendan Momile. Brendan hails from Connecticut, and he obtained his bachelor's and master's degree in biology. He's currently a PhD candidate in plant pathology, working with Dr. Lubberstedt, and his research is centered on understanding transcription activator-like effectors that mediates resistance and susceptibility in bacterial blight of cotton. He's also working to develop a virus vector system for transferring the CRISPR-Cas9 component into cotton germline cells. Welcome, Brendan.
Thank you for that introduction, Catherine. Can everybody hear me okay? Okay, so my name is Brendan Mormile, and today I'm going to be talking to you about the characterization and disruption of tau effector-mediated susceptibility in bacterial blight of cotton. So to begin with, cotton is an economically significant and a diversely utilized crop responsible for the production of fiber, livestock feed, and oil. However, in the past decade, cotton yields have been threatened due to a resurgence in bacterial blight of cotton caused by the bacterial pathogen Xanthomonas citri malvacearum. Now, bacterial blight is a very destructive foliar disease that can affect any aerial plant tissue during any life cycle stage of the plant. The most noticeable symptoms of bacterial blight are the very distinctive water-soaking lesions, which later can become necrotic and ultimately result in premature defoliation and bulb loss. Now, bacterial blight has been largely under control since epidemics in the 1970s due to an increased practice of acid delinting of cotton seeds and an increased use of resistant cotton cultivars. However, in 2011, there was a sudden resurgence of bacterial blight cases across the southern United States, reaching its peak in 2015 with 77 counties reporting cases of bacterial blight. Now, since the most cost-effective and efficient manner of controlling bacterial blight is through the use of resistant cotton cultivars, we have an increased need to better understand the molecular mechanisms and the genetic characteristics that are associated with both resistance and susceptibility. Now, the primary virulence factor associated with Xanthomonas species are TAL effectors, TAL. This stands for transcription activator-like effectors. These mimic eukaryotic transcription factors. Now, each TAL effector has a highly conserved structure. They consist of a type 3 secretion signal, a central repeat region, which can contain 1.5 to 33.5 nearly identical amino acid sequences, followed by a nuclear localization signal and an acidic activation domain. TAL effectors primarily differ within their central repeat region. Each one of these colored blocks represents a nearly identical 33 to 34 amino acids. These sequences differ at the 12th and 13th residue, known as the repeat variable diresidue region, or the RVD. Each RVD has a high affinity for a particular nucleotide, making it so that the order of RVDs within the central repeat region will directly correspond to a sequence within the promoter region of a susceptibility gene. This is called the effector binding element, or EBE. Now, historic US isolate XEM H1005, which was isolated from a bacterial blight infected cotton field in Oklahoma in 1968, has had its virulence highly characterized. It has 12 different TAL effectors, but only one of them, AVR-B6, is solely responsible for inducing bacterial blight symptoms within susceptible cotton. H1005 will inject the TAL effector into the cotton cell using its type 3 secretion signal. The TAL effector will localize to the nucleus. It will recognize its EBE site within the susceptibility gene and cause its upregulation. AVR-B6 specifically targets susceptibility gene SWEET10. Sweet genes encode for bidirectional sugar transporters, and upon their upregulation, results in an increased efflux of sugar being shuttled from inside the cell to outside the cell, the aproplast, where the bacteria lives. This is thought to be a virulence strategy for the bacteria to provide itself with a carbon source. Now, we collected isolates from re-emerging cases of bacterial blight from across Texas, labeled XEM 4, 7, and 9. Now, we figured since H1005 was so well known for upregulating susceptibility gene SWEET10, we figured that these re-emergent isolates may also be able to upregulate SWEET10. However, of our 3 isolates, neither of them were able to upregulate SWEET10, but instead they were able to upregulate SWEET14 A and B. 2 of our 3 isolates were able to upregulate these genes. XEM 4 and 7, however, Xanthomonas 9 was not able to upregulate SWEET14 A or B. Now, our next step was to perform whole genome sequencing using these 3 isolates. We primarily were comparing XEM 4 and 7, which are considered to be virulent. Both of these isolates are able to induce expression of SWEET14 A and B and induce water soaking symptoms in susceptible cotton. XEM 9 is not able to upregulate SWEET14 A or B or induce water soaking symptoms. However, all 3 of these isolates have nearly an identical genome. Each isolate contains a 5 million base pair circular chromosome followed by 2 52 kb plasmids. Each isolate also contains 8 novel tau effectors. These are tau effectors that are not seen in any historic US isolates of Xanthomonas. So we decided to look a little bit closer at the RVD sequences of each individual tau effector within each individual isolate. Since RVD sequences specifically correspond to target specificity and in an isolate's ability to induce a susceptibility gene. So any dissimilarities within the RVD will hinder a tau effector's ability to upregulate a particular gene. Now, we saw that XCM9, the non-virulent isolate, had a high degree of dissimilarities highlighted here in 3 different tau effectors, tau2, tau4, and tau7b. Therefore, we predicted that the primary virulence factor associated with the reemergence of bacterial blight was going to be one of these 3 tau effectors, tau2, 4, and 7b. So the next thing we did was individually clone these tau effectors into a broad host range vector and transform the non-virulent Xanthomonas isolate HM2.2, which is devoid of all tau effectors. We inoculated these isolates into susceptible cotton, extracted RNA, and performed qRT-PCR analysis. We saw that tau7b was the only tau effector of all 8 capable of inducing expression of sweet 14 A and B. Furthermore, it was also able to induce water soaking symptoms to a similar degree as wild type XCM4. Now, in addition to the gain of function analysis, we also performed what's called the loss of function. We created a mutant variety of XCM4 that had a knockout of tau7b. This mutant variety was unable to induce water soaking symptoms. However, the phenotype was partially rescued by reintroducing tau7b back into the mutant. So the wild type variety, XCM4, is able to induce water soaking. We knocked out tau7b. We saw a loss of water soaking. We reintroduced tau7b back in, and we saw that water soaking was able to be rescued. Furthermore, tau7b also has a predicted EDE site on both sweet 14 A and B's promoter regions. What's most interesting is that these predicted EBE sites overlap a ta-ta box. Now, many virulent tau effectors have been seen to specifically target a ta-ta box. The exact reason is not currently known. However, it has been hypothesized that a tau effector will specifically target a highly conserved region within a susceptibility gene promoter. This is to discourage any possible polymorphisms that may occur within the promoter and hinder the recognition and the ultimate upregulation of the gene. Also, it's possible that a tau effector, the ta-ta box, is also capable of recruiting basal machinery, transcriptional machinery in the host cell in order to promote transcription. So it makes sense that a tau effector, which is also trying to upregulate a gene, is also going to target a similar sequence. So in addition to identifying the primary virulence factor associated with a pathogen and its genetic target within a susceptibility gene, we also wanted to develop a method of disrupting this interaction in order to create resistant cotton. So what we did was we used a CRISPR-Cas9 mediated deletion of 7 nucleotides within the EBE site for AVRB6. AVRB6 is the primary virulence factor associated with isolate H1005. So we deleted 7 nucleotides within the EBE site in sweet 10. We performed a tissue culture regeneration, and we saw that the endogenous expression of sweet 10 was not hindered. However, the AVRB6's ability to upregulate sweet 10 was. Furthermore, our regenerated cotton lines were no longer able to showed a significant reduction in water soaking symptoms compared to our unedited lines. So therefore, we were able to ultimately produce a bacterial blight tolerant cotton line by inducing a 7 nucleotide deletion within a promoter. We hope to ultimately apply this technology to the reemergent isolates and induce a similar deletion within the EBE site for tau7b. Now with that, I'd like to thank everybody who's been involved in this project, especially my lab mates and our collaborators at Cornell, also my committee members. And with that, I'll open the floor for any questions that you might have.
Thank you, Brendan, for that great presentation. So we'll open up for questions. If you have a question, please raise your hand for our mic runners. Any questions in the chat? Anybody? Got one.
Was your mutation homozygous or heterozygous in cotton?
Homozygous.
You did get a homozygous. Okay.
Any other questions? All right. Thank you, Brendan.
Thank you.
Next up is our undergraduate Texas A&M oral winner, Katie Toomey. She hails from Houston, Texas, and she has a major in bioenvironmental sciences, minoring in horticulture. She gained some spectroscopy experience in the Colomits lab. And for her undergraduate research, she worked to understand how lipoxygenases affect plant-to-plant communication. And today she'll be talking to us about her REEU research at the University of Georgia, which she tried to find map chlorophyll content in tomatoes. She's also currently applying to different grad schools. Welcome, Katie.
Thank you, Ms. Catherine, for that wonderful introduction. So howdy, y'all. Always wanted to do that. Well, as you know, my name is Katie Toomey. I'm a bioenvironmental sciences senior, horticulture minor. And today I'll be talking to you about the fine mapping of the chlorophyll content locus in tomato. So I did this research over the summer at the University of Georgia in the Vandernapp lab with my junior mentor, Yasin Tapku. And I wanted to say it's a real honor to be here. I'm sure that a lot of you already know what fine mapping is and could probably do this in your sleep. But for me, this was a wonderful experience. It was my first time working in a project so heavily involved in genetics, and I had a blast. So why tomato? Well, besides it being the crop of choice of the Vandernapp lab, it's also a major crop with over 340 billion pounds being produced globally every year. I'm also willing to bet that most of you have seen or ate a tomato within the last week. Today, I'll try to answer, can we lower the region of interest for this chlorophyll content gene? What genes are in that region? And why should you care? So hopefully you can notice the color difference between these two plants. And if not, I provided color bars to the right. So this is something that my junior mentor noticed while working on these stable accessions from Mexico. And he extracted the chlorophyll content and found that while they have similar levels of chlorophyll A, they have statistically different levels of chlorophyll B. So as a Bio 101 refresher, chlorophyll A is the main pigment in photosynthesis, and chlorophyll B is an accessory pigment located in the light harvesting complex, and it helps provide energy to chlorophyll A for photosynthesis. We were interested in chlorophyll B because in both rice and wheat, as chlorophyll B amounts increase, so does yield, and yield is generally something that we're interested in. After crossing these parents, we saw a 3-to-1 ratio with low chlorophyll seeming to be the dominant trait. A few more generations later, and we had these plants to work with for the fine mapping project for the summer. So as you can see, these are the families, and you can see their names on the left and their genomic positions on chromosome 4. While the green means that they're fixed for the high chlorophyll content, and the red means that they're fixed for the low chlorophyll content. The white means that they're heterozygous and will therefore segregate in a 1 to 2-to-1 genotypic ratio in the progeny. Previous fine mapping has showed that our region of interest lies somewhere between marker 712 and 588, and this correlates to a 69.8 thousand base pair region. After selfing the families, we ended up with 48 seedlings per family that we sowed and were genotyped at 13 and 16 days post-sowing. We genotyped them so that we could use them for, we genotyped them so that we could select progeny for marker-assisted selection. And we genotyped them via CASP PCR, competitive allele-specific polymerase chain reaction. And in CASP, there are two dyes, two fluorescent dyes, FAM and hex, that bind to the reference and alternative alleles, they bind to the primers that bind to the reference and alternative alleles, and they'll fluoresce differently in the TCAN reader, and that data is recorded in the TCAN reader and exported to cluster color for our interpretation. So this little gray box is actually what one would see in cluster color. The bottom is the plate, and the top is how the samples will fluoresce. The blue and the green are the FAM and the hex dyes, and you can see how they obviously fluoresce differently, and that means that they're both homozygous for either high or low chlorophyll content, and the red group means that they fluoresce both and therefore are heterozygous. We've repeated this process to be confident in our selection of samples, and we selected 5 homozygous plants per genotype per family. So here are some progeny from family 240, and hopefully you can still see the color difference. And if so, congratulations, you just helped phenotype the progeny. But to avoid going out in the greenhouse with a bunch of paint swatches and trying to do some color comparison, I used a handheld Apogee CCM 200 chlorophyll meter to measure the chlorophyll content indexes of all the samples starting at 25 days post-sowing. And the box and whisker plot on the bottom show that data visualized. So you can see the 5 homozygous plants per genotype, 5 homozygous plants per genotype per family. And so you can see a student's t-test was used to confirm statistical difference in 237, 239, 240, and the parental checks. So because family 237 is fixed at marker 130 and everything to the right of it, yet it still segregates for phenotype, we can assume that the region of interest that causes that difference must lie to the left of marker 130. Similarly, in family 240, it's fixed for marker 588 and everything to the left, yet it segregates, which tells us that our gene of interest must lie to the right of marker 588. Therefore, we know that our new region of interest is 31.5 thousand base pairs. So after we did the actual fine mapping, we took it a step further to do some gene identification. Thanks to the Soul Genomics browser, we have the tomato genome, and we know the 4 genes of interest between those 2 markers. These genes are 280, 285, 290, and 300. So 280 is not believed to be a candidate gene because it's uncharacterized, first of all. Second of all, the SNP for marker 588 is in the middle of this gene, yet it doesn't impede family 240's ability to segregate for it. And additionally, it's not highly expressed in the leaves of tomato. All these indicate to us that it's not likely to be our candidate gene. Contrarily, while gene 285 is also not characterized, its ortholog in Arabidopsis shows, when it's overexpressed, shows paler leaves in the plant, as well as lower amounts of chlorophyll. And additionally, you can see that in Arabidopsis, gene 285's ortholog is actually expressed in the leaves, all of which indicate to us that 285 has a high probability of being our candidate gene. Additionally, 290, as you can see, is expressed highly in the leaves of tomatoes. And additionally, a mutation in its ortholog in rice shows a breakdown of chloroplasts, chlorophyll, and a downregulation of photosynthesis-related genes. All of this indicates to us that 290 is also likely to be a candidate gene. Lastly, gene 300 is not believed to be a candidate gene because, as you can see, similarly to gene 280, the marker for 130's SNP is slightly in side of the gene 300, and yet it doesn't impede family 237's ability to segregate. Additionally, you can see that it actually codes for the same protein as 310, but it actually has a premature stop codon, which causes 300's protein to become truncated, and it's kind of like nature's own CRISPR knocked it out for us. So overall, based on the expression analysis in tomato and Arabidopsis, as well as orthologs in rice and Arabidopsis, we believe that genes 285 and 290 are candidate genes, and currently work is being done in Georgia to knock out these genes independently with CRISPR to elucidate their exact roles in chlorophyll content in tomato. So these are my references, and thank you very much.
Thank you for that great talk, Katie. We're going to do another open question session. So if you have a question, please raise your hand. One right here. Mike's coming behind you.
Very interesting talk. Thank you. I want to ask you if you have sequenced the parental lines for the particular candidate genes already.
I don't know if they've been sequenced. I'm sorry.
Okay.
Any further questions? Back left.
Do you mind explaining the reference and alternative allele concept for the markers?
Sure. So sorry. So we have a reference genome, and when you compare it to our two accessions, the SNPs will either be found in the low chlorophyll content tomato accession or the high chlorophyll content accession. So when we say reference or allele or alternative, what we mean is that the reference is the one that reflects the genome that we already have available, and the alternative is the one that actually has the SNP. So each marker, one of them will have the SNP and one of them will have the reference.
In the back.
Were those markers in coupling or repulsion? Do you know?
I do not know. I'm sorry.
Any further questions? All right. Well, all right. We'll take one more right back here in the back.
Can you explain when you started the project?
So I started the project when the tomato plants were actually sown. So May 29th, I believe. And then I was there for the entirety of the project until these plants were destroyed.
Okay, let's give Katie a round of applause. All right, howdy, everybody. Hope everybody had a good lunch. We're going to go ahead and get started with our next keynote speaker for the afternoon. If you're working your way back in, come on in and have a seat. So our next speaker comes to us from the University of Georgia. Dr. Joann Connor grew up in an agricultural community in Northeast Pennsylvania, and she got her Bachelor's of Science in biology from the University of Scranton, where she began her research with plants. She graduated with her PhD in plant biology from Cornell in 1997, where her research focused on self-incompatibility in brassicas. After completing a postdoc at the University of Maryland, working on floral development in Arabidopsis, she began a postdoc at the University of Georgia in Tifton, where she began her work with apomixis, and she has been researching apomixis ever since then. She currently holds the position of Associate Research Scientist at UGA, where she has held various NIFA, NSF, and Gates Foundation funding for apomixis research. She will be talking to us today about engineering apomixis in crops and giving us an update on the progress towards that lofty goal. So with that, I give you Dr. Joann Connor.
Oh, okay, yeah, they said you'd have to be at the edge to be able to see your. So I'd like to thank the students who invited me or who chose me to give this address. And so I'll just get started. Okay, there we go. So what is apomixis? Apomixis is asexual reproduction through seed. What this means is that the progeny of the apomictic plants are genetically identical to the mother. And this is just a picture of pennisedums from seed that are from an apomictic parent. Apomixis is actually found relatively frequently. It's in 400 species, 35 different families. Now, even though apomixis has this really non-complicated definition, it's been around a long time, and what plants have learned to do is use multiple pathways to actually become apomictic. And so there's not just one apomictic pathway. There's 6 that we know of if you count whether or not endosperm is autonomous or you need pollination. So I do want to give you a little bit of a background on apomixis. So if you have your sexual pathway at the top, and you have your megaspore mother cell, you go through meiosis, you have your megaspore that divides 3 times to make your 7 or 8 nuclei, 7 cell with, you have your egg, your synergids, your antipodals, and then your central cell, which will end up being pollinated to make the endosperm. And then you have fertilization by the pollen, and you get your nice sexual recombined embryo along with endosperm formation and a healthy seed. So in citrus, they do something that's called a sporophytic apomixis. And what happens is you actually have to have the sexual pathway for this type of apomixis. And as it gets close to when pollination is going to take place, you start seeing these little embryos forming from the nucellar cells. So they just start to divide and they become embryos. The one problem for engineering sporophytic apomixis is that you usually end up, so you have to have the sexual embryo sac pollinated to get endosperm, because apomictically you're only making the embryo. And then you're going to have a competition between this sexual embryo and your one or multiple sporophytic apomictic embryos. And this usually leads to actually having multiple seedlings within one seed, which is not what you want penetrance-wise. You want it to be all apomictic. And then you have another way, it's called diplospory, and there's 2 types of apomixis that are gametophytic, and gametophytic just means that it makes an unreduced embryo sac. And then that unreduced embryo sac then becomes the embryo. And again, if you're diplospory, the megaspore mother cell actually doesn't go through meiosis. But you make an embryo sac that looks exactly like the sexual embryo sac. So people who work on diplospory have a hard time early stage to tell whether or not their plant is apomictic or not. And then again, you sometimes have pollination of the endosperm. Some apomix actually just start creating their own endosperm, and you have unrecombined ploidy level the same as the mother plant. And the final type is apospory. And with apospory, what happens is that if you're a strong apomict, meiosis doesn't take place, and the only embryo sacs you see are aposporous, meaning unreduced, non-recombined. And in our genotype or our species, it actually doesn't have antipodals, so it's very easy to be able to distinguish sexual embryo sacs from apomictic sacs. Again, you have parthenogenesis, which means fertilization, or you don't have fertilization of the egg cell. It becomes an zygote on its own, and then it becomes a clonally propagated seedling. I usually like to give a picture of real life, what this looks like. And so when you start making your phosphorus initials and then you make your unreduced, unrecombined embryo sacs, that's the process known as apomeiosis in apomixis. And this is Sancris ciliaris, and we don't know what triggers the phosphorus initials to start swelling up, which is really how you can just tell them different from nucellar cells. And then they go through 2 divisions, and we don't have antipodals. If you're clearing here through the with methylsalicylate, usually you can't see the egg cell, but you can always see the polar nuclei. And then the next step in apomixis is actually turning the unreduced egg into an embryo, and that we call parthenogenesis. And here is just in purple is the multicellular embryo. This is 2 days after what would be natural pollination, even though we don't allow pollen to fertilize, so you don't have endosperm formation. All right, so we've been genetically in the Pennisetum sancris species. Apomixis is basically 50 megabase loci, and it's non-recombined. We get low recombination in sancris and almost none in Pennisetum squamulatum. And one of the reasons why is that in squamulatum, you have, this is the ASGR, and when we look at pollen meiosis, the squamulatum chromosome will, they'll pair up, and then they get to this repetitive area, and then no pairing takes place. So we really don't think we'll ever be able to get smaller, a smaller region for the locus in squamulatum. In sancris, every once in a while, we can find a plant that recombines where we can separate out parthenogenesis and apomeiosis. We know parthenogenesis is located in this area on the UGT197 back, which is in the low copy area. And we know the apomeiosis is somewhere in here, based in the high copy region, which makes attempting to walk in this locus almost impossible. The other thing we do know is that this repetitive flock is only found on the ASGR carrier chromosome in squamulatum, but in sancris, it's on these high repetitive repeats are on every chromosome. So why do we want apomixis in crop plants? And the reason is it would allow you to maintain hybrid vigor in the F1s without the need for annual crosses. So this is just a very basic scheme of how it would work. You have your 2 parents that are inbred lines, and when you cross them, they make your F1 hybrid, which is uniform, more vigorous, and better yielding. However, if you allow that really good F1 plant to self, and then you plant out those seeds, what you're going to do is get all the segregation within the parental lines, which will lead to ununiform growth and not this uniformity, and which is what farmers really like. However, if that F1 hybrid was apomictic, then all the generations after that F1, when you planted out the seed, would look the same. So when apomix, there was a thought at one point that breeding with apomix was a dead end. Like, okay, you're apomictic, there's nothing more you can do. And that's kind of true on the female side, because if your progeny are all maternal clones, you can't do anything with it. But what they did find, breeders found, is that they can actually use the pollen and bring in apomixis to create apomictic lines that are actually out in the field. And this has mostly been used in forage grasses Berkeria, Pispalum, and Poa. And so what you do is the first thing you have to breed with an apomictic, first you have to know, are they apomictic or not? And it helps if you're a very strong apomictic. Apomixis sometimes isn't 100% penetrant, and it's called facultative. And so therefore, when you make the cross, the offspring will not always be apomictic. You have sexuality and apomixis within the same plant. Again, so if you're trying to use this as an engineering for crops, you do not want to be facultative. You want your penetrance up very high. However, you can use it to breed. And so one of the things that once you know whether you're apomictic or sexual, your plants, and now pretty much everything's done by molecular markers on whether a plant is apomictic or not, or has the locus or not. One of the problems is that your sexual plant, usually it's easiest if it's the same ploidy level as the apomictic in terms of making your cross. And that's where problems come in, because a lot of times the apomictics are highly polyploid, and your sexual plants within the same species are usually diploid. And so a lot of times they'll take the diploid plant, make it a tetraploid, so they can more easily cross in the apomictic genotype. And so, but once you make this cross, you can find your best phenotypes, and if they're apomictic, start your replication tests, and then once you know the variety or the plant is you think it would be good, you can get it out into the farmer's hands. All right, so we can breed with apomixis, mostly for the forage grasses. However, there are no food crop plants that are apomictic. And so what they've tried to do in the past is to actually integrate in apomixis from wild species. So there's been work done mostly on these three species. So in maize, they try to bring in apomixis from Tripsicum. For pearl millet, we tried to bring it in with Pennisetum squamulatum. And in wheat, they used Elamis. And what happened was that they made the first cross, and it looked really good. Now, fertility wasn't great, but the seed that was produced was highly apomictic. But then you have this wild species, and then you're cultivated, and so you have to start trying to get in the back crosses to make it so it's more cultivated and not so wild. And what they found is that as the generations go by, your ability to be apomictic, to make clonal seed, just starts decreasing and decreasing and decreasing, and you end up with a lot more sexuality. So I wanted to show you in pearl millet, which this was done by Wayne Hanna at the University of Georgia. So our PS26 plant has 56 chromosomes, and he was able to, with a bridging species, get to a backcross 7 line, and that plant had a good proportion of aposperous embryo sacs formed, 93%. Our pollen viability wasn't too good, just 37%, but it was enough to, set the few seeds that we got. So our backcross 7, we had 6.5 seed per centimeter, whereas the wild type tetraploid has 80 to 100. So again, in terms of fertility, when we kept it being clonal, then fertility went down a lot. And our back cross 7 ended up having 2 chromosomes. One that with the green is the ASGR carrier chromosome. And then this is a squamulatum, or our bridging species, that was used in the cross. And so then when we got to a back cross 8, where there was only one, the ASGR carrier chromosome, and all the other chromosomes were tetraploid promillet, we increased our, where are we? Here we are. We increased our seed set. We're still nowhere near up to wild type. The problem is now these, we've decreased our ability to be apomictic in a lot of our ovaries. And so that increase of seed set was basically based on sexuality, which isn't what we want. So if it doesn't look like we're going to be able to easily integrate in apomixis from wild type species, what's our next option? And our next option is to engineer. So for me, we're not there yet, but what would be the easiest design is to know the genes that a natural apomix use to do apomeiosis and parthenogenesis, and put that in as a cassette and then put it into crop plants and become apomictic. We have a problem with that in that right now we have 2 parthenogenesis genes that have now been cloned from natural apomix, the ASGR baby boom like gene, and they call it the parthenogenesis or the PAR gene from dandelion. And right now we have no confirmed apomeiotic genes. So we still don't know what the natural apomix do to not go through recombining, not go through meiosis, or to make the aposporous embryo, or the aposporous initial to make the unreduced embryo sac. The other thing that we don't have, since most apomix are polyploid, is we don't have a good genome sequence for any apomict. So these are wild species that weren't high on the list to be sequenced early. Right now, JGI, is sequencing a lot of our apomictic Sancris and Pennisetum species for us. We're hoping technology has become good enough that we'll actually get chromosome assemblies. Part of me says that's not going to happen because we have so much repetitive DNA within these polyploids, but let's go bioinformatics. I hope they can do it for us. And to think about, and squamulatum, they have to be able to give us a whole complete ASGR. It has to be 50 megabases, completely linear. So I'm hoping, but we'll see. The one other thing is since most apomix are polyploid and they go through all these different pathways to become apomictic, we know that the baby boom, the parthenogenesis genes are 2 different genes. They're not the same. They're both transcription factors, but they're not the same transcription factors. So the one thing that we don't know is what's going to be the best apomictic pathway. So what is wheat going to like the most? What is corn going to like the most? What is rice going to like the most? Will it turn out that eudicots and monocots actually will use the same system, or will one over the other be more effective? So it doesn't mean that we can't get apomeiosis, though. This was first done in Arabidopsis and then in rice. And what they did is they actually engineered apomeiosis by using meiotic genes. And so remember, when you're apomictic, you have to be unreduced and non-recombined. So you have to get your segregation. You can't have your segregation in your second division in meiosis, and you have to stop recombining. And so they know that if you add the SPO, the REC8, and OSD1, all have to be in mutants, in a mutated form, so they can't be functional. Then what you'll end up with is an unreduced embryo sac. You'll also have unreduced pollen, which is a little bit different. At least for our apomictic plant, the pollen undergoes meiosis correctly, meaning it becomes reduced. Not in other apomictic plants, pollen is at the higher ploidy level. And so the other, I find it very amazing, is that the penetrance of this triple mutant is 95 to 98% in Arabidopsis and rice, which, that's really good in terms of being able to actually use it to engineer something and then get it out to market. There is one other, well, there are a couple other meiotic mutants that people are thinking about trying to use to engineer apomeiosis. One is dyad, which was found in Arabidopsis. It's a female allelic meiotic gene. The problem with dyad is right now it only makes 0.2% of your female gametes are functional. So it's just, again, the penetrance isn't high enough. And that's more like the mime is more like diplospory, in that you're starting at the megaspore mother cell. As for aposporous embryo sacs, right now there are mutants that look like they form aposporous-like embryo sacs, and they're part of the small RNA pathway. The argonauts, the RNA-dependent polymerases, the suppressors of gene silencing. And in all cases, these mutants have reduced fertility. What's interesting is some of these mutants actually work in a heterozygous form, which in natural apomix, you're usually more heterozygous than homozygous. But again, the penetrance is the problem to actually get it as an engineered for engineering. So before CRISPR-Cas9 technology, the only way to make these triple mutants was to find the mutations in the genes you were interested in, keep them in a heterozygous form, because as a homozygous form, you're either lethal or you're increasing your ploidy level, and you get it all together. And the Mercier lab actually did this in rice, but I wanted to just show you the steps that was required for them, all the crossing and the genotyping they had to do. And every time they made the cross, less and less of their plants are what they needed to make the triple mutant. So there was a lot of, they had to go through a lot of plants. But as you got to step 3, 1/64 of your plants would actually have the mime phenotype, meaning you've mutated all 3 genes, and you got them all together based on crossing. So kind of a bravo to them for being able to do that. One of the things I think will totally revolutionize the ability to actually make apomixis in crop plants is the CRISPR-Cas and now the associated technology. Because now you can mutate those 3 genes all in one shot. So you don't have to worry about all those crosses and the lethality. And the CRISPR technology, while it's easier in a diploid, they have shown that it works well in polyploids. So they've been actually able to knock out like an octoploid genome. They found mutations in all 8. So this will really change what we're able to do in agriculture. So basically, even though I know probably most of you have that, about the outline of CRISPR, but I just wanted to You decide what genes you want to mutate, and then you have to construct your vectors. And usually you have to validate by doing an activity validation. And then one of the big things is that right now you still have to have a good transformation regeneration system to use the CRISPR-Cas. Hopefully that will change in the future. We just have to wait and see. Once your plants are regenerated, then you start looking for your mutations, and you hope to find that plant that has all the mutations in a homozygous form, so you can start analyzing the phenotype. This is just because the technology is changing so quickly. You know, it started where they were just finding indels. Now you can actually create base pair substitution. So if you want to change one amino acid, you can, or create a stop codon. You can also target your insertions. You can target deletions. So to guide RNAs, you can delete almost the complete gene so you know that you're totally getting rid of it. And the one thing that I'm really interested in is that now in Arabidopsis, they can do chromosomal translocations. So we worked for years trying to get that squamulatum ASGR locus off the pennisetum carrier chromosome and onto a millet chromosome. We irradiated, well, mostly irradiated, but we screened and screened and screened, and we could never get it translocated onto a pearl millet chromosome. It would be really neat if we moved it to see if it would become more functional if it was actually on a pearl millet chromosome instead of the squamulatum chromosome. So pretty much with the MIME, with it being such high penetrance, we have apomeiosis figured out. So the next thing you have to do is you have your unrecombined, unreduced egg, and you have to then get that egg not to be pollinated. You either have to knock out a set of chromosomes or have parthenogenesis. So I'm doing this a little bit in history, and the first people to show that you could, well, not the first, but to show that you could eliminate a genome was through SNH3. And it was done in Arabidopsis to start. And what the SNH3 does is that you create a weak and a strong centromere. And I mean, and this has been used to create addition lines. But here, the weak centromeres, basically, if a weak centromere is is always as a weak centromere, the plant is healthy and fine. So you can be weak, but as long as you don't get a strong centromere in with you, you're a fertile plant. But what happens is if you have a strong centromere, when you start doing your divisions, only the strong centromeres are being put into the new cells, and the weak are basically being left behind and lost. And so if you have in an egg cell, if you have your weak and your strong centromeres, you have fertilization, and then as the zygote starts dividing, you slowly lose those weak centromeres. So for SNH3 to work in apomixis, you always have to have that cross, or you have to have a way of having weak centromeres only in the egg cell. So like many genes, they've found enhancer genes. So they were able to make clonal, they used SNH3 to make clonal offspring in Arabidopsis. Penetrance was a problem, as was, you would have aneuploidy, where one of those weak chromosomes would hang around. And so you didn't get rid of all the male component. They've used the SNH3 to engineer in maize. and they get about a 5 to 8% haploid induction. And in the most recent paper on the SNH3, in their discussion, they put designing an efficient haploid induction in each species may require different SNH3 modifications, as different constraints could apply to the function of SNH3 variants. And then they go on to say, we may be better off to try to directly remove SNH3 to create the weak chromosomes. But what that is showing is that that when they've tried to move the SNH3 into other species and into other crops, what worked in Arabidopsis doesn't necessarily work in rice, or what has worked as well in Arabidopsis doesn't work as well in rice. And that goes back to where I was talking about, do we know what apomictic pathway the different plants are actually going to, our crop plants are actually going to work the best? All right, so they have found additional genes, but to do genome elimination or haploid induction. And just recently, in 2017, they found the gene that is responsible for a lot of the haploid induction in the stalk 6 of maize. And it ended up being a pollen-specific phospholipase gene, which my guess is when they were like, when they were trying to walk into figuring out what gene was causing haploid induction, I doubt many scientists said, oh, I bet it's the phospholipase gene that's doing it. And they have found in maize that different phospholipase genes can also be used for haploid induction. And there's also a gene, DMP, which again is not a phospholipase, but when you add that in with the matrilineal PLA1 or not like dad, basically 3 papers came out within a month of each other, all showing that this gene was cloned and was the stalk 6 haploid inducer. So right now, they've used in CRISPR, the matrilineal gene is found in monocots, and so they've mutated it in multiple monocots, and like wheat, rice, and foxtail millet, and basically haploid induction is less than 10%, which is similar to what it induces in maize. But in terms of engineering, we're not up to where it would need to be to use it to actually create apomictic crop plants. So there's also so that's the genome elimination either through matrilineal or modified SNH3s. And then the other way of doing it is actually parthenogenesis, where your egg starts dividing into a zygote. And what we now have 2 parthenogenesis genes. The PAR gene from Taraxacum was, the paper was published less than a month ago, I believe. Again, talking about CRISPR-Cas9 and what it can do. They had a 300 kb region that they knew the parth gene was in. And natural apomix usually have low, their loci are regions of low recombination. So you usually have a big area to have to find your gene in. What they did is they had 13 genes on that 300 kb. And because Taraxacum can be transformed, they just took those 13 genes and did CRISPR and knocked out the 13. And then they found the gene that didn't show parthenogenesis, which was a lot easier than trying to overexpress the gene or so it's easier to stop a to stop something from happening, to verify that that's the gene of interest. It's interesting that it's a transcription factor. And what's even more interesting is it's very similar to the baby boom gene in that it's the early expression of this transcription factor in the egg cell that leads to the zygote being formed. So for the longest time, we thought, or at least I thought, that it was probably a mutation or a change of the protein itself that was causing apomixis. But it's actually the timing of expression, at least in these two parthenogenesis genes that we have found and shown to be parthenogenic is what's happening. So I'm going to talk a little bit more about the ASGR gene, because that's what we work on, baby boom gene in the lab. It was 2015, I think, when we proved that it would induce parthenogenesis. But before that, we found that it was on an ASGR back clone that we had sequenced. It had similarity to the baby boom gene in Arabidopsis, where if you overexpress it in leaf tissue, you get somatic embryogenesis. So it has the 2 AP2 domains, 8 exons, and what really made us focus on this gene was that we found that it was transcriptionally active in ovaries at about the time we would expect parthenogenesis to start, which is about a day before natural pollination. And we found it in all our 12 apomictic species or apomictic Sankara species, but none in the sexual. So that made it a good candidate. This is just showing that it's the promoter, and this is transformed promillet. We cannot transform our wild species, Sankaris or Pennisetum. But promillet is very closely related, and we crossed squamulatum into promillet. So when we did promoter Gus, we see that it's on in the egg cell. We're still not sure about the synergids. We think that might be leakage of the Gus because you have triton in the tissue. And then our ASGR baby boom gene, which is the apomictic one, continues on through the embryo development. This is just a confocal picture to show that in a sexual embryo sac without the transgene, you have your egg and your polar nuclei. If you have the transgene, you start seeing embryos formed without pollination. And that this occurred both in our T0 and our T1 plants. This shows that we can reduce a tetraploid or any kind of polyploid down to a haploid level. So we started off as a 4N in the T1, we went down to a 2N, and a T2, we went down to an N. Promillet does not like being haploid. Those seedlings, I mean, they got about that big, and then they just stopped growing. They're not happy. So while it's nice that it worked in promillet, we really wanted to see if it would work in rice and maize, because we weren't sure that the ASGR promoter would work in rice and maize. We switched over to the Arabidopsis DD45, which is an egg cell specific promoter. And this just shows that it doesn't matter what construct we use. It can be cDNA, it can be genomic, it can be our ASGR promoter, or it could be DD45. But in all cases, we are seeing haploid production in both, and the haploid, the H stands for haploid. And we don't see it in wild type, but we do see it in all the transgenic lines. All right, so of course there has to be a problem, because if this was totally working, things would be further in the pipeline. And like the SNH3 or the matrilineal, our problem is seed set. And then our problem is embryo development. So this is actually a haploid producing maize line, but most of the ovaries are not producing seed. But in here, in these few seeds that are produced, there are haploid plants. And with our rice, this is a wild type. So this is seed where we know that endosperm is being formed. But when we start looking at the development of the embryo, you can notice we can get stuff that doesn't even look like an embryo. I think it's just cell proliferation. And embryos that sort of have a shoot apical meristem and a root, but they're definitely not wild type in shape. The other thing about our baby boom transgenes is we have tried to do it in U dicots, and in tobacco we get less than 1% parthenogenesis. And in Arabidopsis, I have never been able to get parthenogenesis. So other labs have created apomictic rice plants, and they've done this by combining either baby boom genes or matrilineal lines and the MIME strategy. And actually, just recently, there's been multiple review papers about synthetic apomix, and in a couple months, there's probably going to be 3 or 4 more out. So if you're really interested, those are coming out soon. And so we have, apomixis, synthetic apomixis has been created, which is great, using MIME and, like I said, either baby boom or the matrilineal. The problem is penetrance, and the penetrance is usually, the issue is based on the parthenogenesis. So that's where the real work has to go to. And one thing I noticed when I was reading, the various papers is that a lot of times we don't talk about seed set. Now, the people who wrote the fixed paper, they were really good because they actually put their seed set against wild type and then how many of those seed were clonal. And so they have 0.41% clonal seed. And most of that is based on parthenogenesis not going well. Or since they use matrilineal, it would be haploid induction. All right, so I was when we first found baby boom, we looked at the phylogenetics of the gene. And in dicots, it's pretty, usually it's a single or there may be 2 baby boom genes in the genome. In the monocots, we had more baby boom-like genes, and they seem to actually go into clades. And so when we first started this, we were thinking that maybe there was something specific about the protein, the baby boom proteins within this clade 1, because the 2 genes shown to induce parthenogenesis, the rice baby boom 1 and R. squamulatum, claded together. The other interesting thing, maize and sorghum, both which aren't apomictic, didn't have a baby boom within the clade 1s. So what we did was, and rice actually has 4 baby booms. So one of the diploids that has been sequenced, foxtail millet, Ceteria italica, it actually had just 3 baby booms in its genome, and there's a baby boom in each clade. So we thought this is the perfect baby boom genes to use to try to figure out if The clade 1 is very specific for parthenogenesis, or if all the baby booms could do it. And it turns out all the baby booms can do it. And at this point, we really don't see one of the baby booms being better than another. So as long as you have a baby boom gene, it seems as long as you have a baby boom gene that is expressed in an egg cell, you can get embryo development. And at least in rice, you can get haploid seedlings. Again, penetrance, seed germination is all over the place within the lines. And also how many haploids you get, and fertility is very variable, which leads me to think it's expression. That apomix have had millions of years to get the ASGR baby boom expression exactly right. And when we take it out of squamulatum and try to cross it into pearl millet, and we're changing the genome and the methylation status, and that we are changing how these genes are being expressed. And we're going to have to really work on finding that perfect expression pattern to be able to engineer apomixis. All right, so just since I'm out of time, a few quick thoughts. So again, I hope I've shown you that the penetrance, especially when you're talking about genome elimination, haploid induction, or using the baby boom or the PAR gene, that really seems to be the sticking point right now to getting out crop plants that are actually highly apomictic. The one thing we're assuming that once we make these crop plants apomictic, that the hybrid vigor will stick around. And we haven't shown that yet. I sure hope they stay hybrid vigor, but we don't know. Can MIME be used in all the crop plants? So it's really good in rice, and it's really good in Arabidopsis, but we don't know if that's going to keep in other crops. Again, the same thing, we can't get baby boom to work in eudicots. Will the PAR gene work better? One thing that amazes me is that there's a lot more going on in embryo development than when I first started working on this than I thought. The species just are not the same. And the last thing, is if you think about it with the apomix, you're changing your endosperm ratio, both maternal to paternal, and also your clonal seed or your clonal developing seedling to the endosperm. And right now we don't know how big of a factor that is and why we get low fertility. All right, so very quickly, NSF, NIFA, and the University of Georgia, along with Gates, I haven't really talked about, has given money for this research. Right now, the lab is pretty low. I actually work in Peggy Ozias-Aiken's lab. I'm a research scientist. And in 2021, we graduated 2 graduate students. Lovepreet did the work on the other baby booms from Cysteria. Gurjot, we actually can induce haploids using synthetic chemicals. So we can talk about that tomorrow or whenever people are interested. And then just lots of people throughout the year who have worked on this study. I mean, we're talking 25, 30 plus years now of research. Thank you.
Thank you, Dr. Connor, for that excellent seminar. So we're going to open up for questions now. So if you have a question, please raise your hand and one of our mic runners will get to you. If there's a question in Zoom, please raise your hand and we'll make sure we get your question read. So any questions from the crowd? Back, right?
Hey, Dr. Connor, I have a question.
Yeah.
So say we get like functional apomixis in a hybrid crop to work. Do you think seed companies would ever give you any like pushback on adopting that due to concerns over seed saving, maybe?
No, because it will save them not having to do the annual crosses. And the truth of the matter is farmers in the US are not going to store their own seed. It's just, there's too much variability in not getting good germination rates. So they can sue the company if they get horrible germination rates. If they store it, they're not going to. Where it could potentially really help is to get it out in where you do the small farming communities. So that's why, no, I don't think there's going to be too much of a kickback.
Becklin.
Thank you very much for very good presentation. One of the basic assumptions for this work is if we get a apomictic hybrid, it will be as high yielding as a normal hybrid. So what is the evidence for a normal seed set in apomictic type versus non-apomictic normal type in wherever we have apomictic things already going on? Because if apomictic is abnormal compared to the reproduction thing. So if apomictic version is less yielding, less seed producing, then the whole exercise will be counterproductive. Thank you.
Yeah, and you're right. And that's one of the really big questions that we are assuming that if you can create an F1 hybrid that was highly vigorous, just, creating it by crossing, that then if you make that F1 apomictic, we're going to keep that going year after year after year. As for the apomictic plants being vigorous, they're so heterozygous. And, most of the ones that we work with, it's more the consistency. They always have this much seed set. They always grow this big. But yeah, it's an assumption that apomictic crop plants will maintain that hybrid vigor.
My question was not for forage production or biomass production, but for seed production. When we're looking at the seed crop, we are interested in the hybrid maize, or even there is a work going on in Australia funded by Bill Gates Foundation. You might be knowing Dr. Quiltinao.
I can't talk about that.
Yeah, but the idea is that if we engineer apomixes, there will be possibility of hybrid cowpea or hybrid sorghum or hybrid this seed production. My basic question is apomictic version as good in seed production as a normal? That's the basic assumption. For forage crop, I think it will work. Thank you.
Yeah, and I guess all I can say is they really don't know because we don't have any apomictic seed crops. So our wild type apomix, they're not really, we're not using them in any kind of production for seed. And so, and one of the variabilities that the Gates, I can't say this, that they're trying to figure out is, and it gets complicated, but how you could create apomixis, but keep components separate and bring it together so that you can make all these variations that a breeder wants to be able to do, and then let them stay apomictic. So we're thinking about that. It's just a very complex engineering feat that will need to happen. And I think the fixed lines are actually hybrid rice lines. So the paper did not talk about multiple generations of the fixed lines, but I'm assuming once they can get a couple generations of the fixed line, we will at least know in rice whether that fixed line continually gives good yields. But like, we're not there yet.
I think we have time for about one more question. If we got one. Mike's coming behind you there.
I wish we were.
Yeah, maybe you could state it another way, or some of the thoughts you've expressed, just how easy do you see in the future when this is, if we do get it working, how easy will it be to maintain an apomictic restore, if you will, or an apomictic line? Because obviously you'd want to have one of your parents to be able to confer that apomictic in the hybrid cross.
Pessimistic side of me says it's going to be super hard to do. Non-pessimistic side of me says technology is changing. And we are going to need very specific promoters to be able to get this to work in like a crossing line. And that I don't think we have yet, because it would really be best if we could have just a gene that's on in the egg and then gets turned off after it's done its thing. And I think that is going to be hard to find. So we have theories on how to do it. I just don't know how practical it's going to be to actually be able to do it. But that's the fun thing about science. You try.
Great. Thank you, Dr. Connor. We're going to wrap up questions there. Let's join me in a round of applause for that wonderful talk. So we're going to be beginning our next section here. We're going to be having some of our travel scholars coming up to speak. Also, as a side note, I believe we have some coffee refills coming sometime soon. So if you need a caffeine boost here in a few, that will be coming. So Fabian's going to come on up and do our next intro.
Okay.
All right, so our next portion of the afternoon is 3 outstanding scholars that we found. We combed the nation and found these 3, awarded them a travel scholarship sponsored by Corteva Agriscience, and flew them down here. So our first speaker is joining us from Geneva, New York, and Cornell Agritech. He'll be speaking on sex determination and dimorphism in shrub willows. So please welcome Brennan Hayden.
All right, thank you. And yes, as Fabian mentioned, I'm from Cornell University, and my research primarily focuses on understanding sex determination and dimorphism in shrub willow for bioenergy. But today I'm going to be talking about a unique monoecious line that we have in the species and how it can really serve as a unique and novel genetic resource. So a little bit of background about the system I work on. Willows are the genus Salix, which has over 300 species, and these include the tree willows, which you're probably most familiar with, as well as shrub and dwarf willows. And shrub willows are the section Vitrix. And these have applications as ornamentals in bioenergy and in ecological restoration. Although in the SMART lab where I'm located, we're primarily focused on genetics and breeding of shrub willow for bioenergy usage. So in bioenergy, shrub willow is planted in high density settings as shown here. And it is encompassed every 2 to 4 years, and those wood chips can either be used directly as a feedstock for biofuel or burned to generate heat or electricity. And so these typically have around a 2 to 3 year generation time, which presents some challenges with breeding and genetics. And my research focuses on the species Salix purpurea as a model system for the shrub willows. And the shrub willows are dioecious. So in the left up here, these are the female flowers. We call this a catkin, and on the right are the male flowers. And so these have a ZW sex determination system. And all that means is that the females are ZW and the males are ZZ. So the W is only inherited through the female line. This is the same type of sex determination inheritance that is observed in birds, including poultry. And then willows are also a close relative of the poplars, or the genus Populus, which are another important dioecious bioenergy crop. So some of the things we learn about sex determination in willow can be broadly applied to the poplars as well. So one of the first things I wanted to do in my PhD research was to sort of characterize these monoecious willows and how that sort of varies across environments. So this is a picture of a monoecious catkin, and you can see sort of the upper part of this catkin, these are male flowers, and the lower part here are female flowers. So in this monoecious willow, we have catkins that are mixed, male and female, but the amount of male or female flowers and the pattern varies highly. So we wanted to see, are there environmental effects behind this, or is it totally random? So we had 94003 is the name of a monoecious line we have in our collection. And this was found at 3 different locations. So at Cornell Agritech in Geneva, New York, at the Lake Erie Extension Laboratory in Portland, New York, and then at West Virginia University in Morgantown. And these were in randomized block designs. And so we actually looked at the percent maleness of catkins in the field to see how this varied between environments. We also took a subset of shoots 2 months early in the winter and forced them in the greenhouse from the Geneva site to see if early flowering could affect percent maleness. And turns out it didn't. So that likely means that the sex of the flowers, or the relative ratio of male to female flowers, is determined at least a few months before flowering time. And so we created a general linearized mixed model following the beta distribution to model the percent maleness, and that equation shown here. And we modeled the actual field, so which of those 3 locations as a fixed effect, and then the plot as well as the shoot within the plot as random effects. And we found that all 3 of these had a significant effect on percent maleness, which suggested to us that there are both macro and micro environmental effects controlling the relative proportion of male to female flowers. This is not entirely unsurprising. We see that the environment has effect on male to female flowers in other monoecious species like cucurbits. And this box plot here shows some of the distribution of those data. And so we can see here the mean between whether they were forced in the greenhouse or analyzed in the field in Geneva was not significantly different, although the variation was higher in the field. But there are pretty strong differences between the 3 locations. And the gold here just indicates that when we measured percent maleness by catkin, and the red when we measured it by shoot. But one key thing to note from this is the range of values. So in most of these, we see the ranges from 0 to 100% maleness. So we can find entire shoots on monoecious plants that will be all female or all male. And that suggests to us that there's a possibility or a risk of misidentifying monoecious plants as male or female, particularly when there's only a few catkins, a small catkin sample size. And this will be important in the next slide. So another thing that we wanted to do with this monoecious plant is to see if we could generate self-crossed families, right? So willow is dioecious. It is an obligate outcrosser. So there's nothing reported to date of actually generating a self-crossed family because these are dioecious. So in 2015, we selfed 94003, our monoecious parent, to generate an S1 family. In this family, we had over 500 progeny. So that alone was quite novel, that this plant was viable and produced viable progeny. And there were monoecious plants in this family, which also told us that monoecious is heritable. So we took one such monoecious individual, which we're calling Z, and then we selfed that to generate the S2 family, 18X551. And so we have 3 years now of flowering data on this family. And this pie chart here shows the distribution of those flowering data. And it looks to be segregating for about 2/3 monoecious and about 1/3 male. There are a few what look like to be females in this family, although we suspect that those are false positives. All of them only have 1 year of data supporting that they're female and only had a few catkins. So again, they're likely monoecious plants that we just didn't have a large enough catkin sample size to identify them as such. We also collected GBS sequencing on every individual in this family, flowering or not, only 1 year after planting. So before we could get much mortality in that family. And when we looked at the sex determination region in these GBS data, it appeared to be segregating for ZZ and ZW. There did not appear to be any WW individuals, which suggested to us that that is likely lethal. And furthermore, the ZZ individuals corresponded to males, which is what we would expect, and the ZW individuals corresponded to monoecious. So that led us to hypothesize that Monishi is inherited on the 15W sex chromosome, such that plants that are ZW are Monishi-sensitive female. And this figure here sort of shows our hypothesis for that inheritance. So, we think, ZZ are male, ZW are monoecious, so we're calling this modified W a WH such that it results in Monishi instead of females. And then that homozygous WH is lethal because we didn't find any individuals in over 400 progeny that were WW. Now, to confirm this hypothesis, we decided to outcross our monoecious parent to our reference male and reference female individuals. So when we outcrossed it to our reference male, we now have 2 years of flowering data on this family, and we indeed see about half male, half female, which is what we expect if we cross a ZWH with a ZZ. We expect 50% ZZ, 50% ZWH. There is a slight skew of males here, although that's likely due to only a year or two of flowering data. So there are almost definitely some false positive males in here, but pretty close to 1-to-1 after only 2 years of data. And when we outcross it to our female reference individual, things get really interesting. Again, we think that WW, or we thought that WW was lethal because we don't see any individuals in our S2 family that are WW. But this family is over half female, it looks like. And if WW were always lethal, we'd expect a 1-to-1-to-1 ratio of male, female, and monoecious. But instead, we see about a 2-to-1-to-1 of female, male, and monoecious. So that suggested to us that WWH, so our normal W from our female parent and our WH from our monoecious parent, that is actually viable and female. So that suggested to us that perhaps the WH, this modified W, it has a large deletion such that it disrupts both a sex determination gene and some gene critical for plant function. And so to confirm this, we're currently in the process of performing resequencing. So we're collecting PacBio data on our 94003 monoecious parent to get an assembled reference genome, and then Illumina short read sequencing on a subset of males, females, and monoecious progeny from these two families, as well as that S2 family. And we're still, playing around with the 94003 assembly, but so far the data appear to be confirming our hypothesis that indeed there is a very large deletion, almost a megabase, on this modified 15W that is indeed is absent several candidate master regulator genes for sex. And so again, this is just to summarize our hypothesis of the inheritance of Monishi, that Monishi we think is on this modified W called a WH, such that the progeny are monoecious, and that homozygous WH is lethal due to this large indel, but that a WWH is viable and female. Now, all this about monoecious plants, this is interesting, but why do we really care about monoecious willows? Well, one of our original goals behind generating this S2 family was to obtain inbred progeny so that we could perform some genetic mapping studies that are otherwise difficult to do due to the highly heterozygous nature of willows. And so this family is still relatively young, only a couple years old. So right now it's in single plant plots. So we chose to look at high heritability leaf dimensions traits. So leaf dimension traits like area, width, perimeter, these are all have been shown to have a high heritability in willow and also be correlated with biomass yield. So they're interesting traits. And even looking at single plant plots, we identified multiple QTL for many of these traits. And within many of these QTL are interesting candidate genes. So notably on chromosome 16 is a PIN1 homolog, and on chromosome 17 is a Kanadi homolog. And in Arabidopsis, these genes actually interact to specify the abaxial, adaxial leaf identity. So these are strong candidate genes in leaf morphology, and indeed, both of them are showing up in our QTL confidence intervals. So that really suggests to us that this family could be very powerful for future genetic studies. And so some of our future directions, as I mentioned, we're performing resequencing on some of those monoecious progeny to narrow in on the actual causal locus of Monishi. And then this spring, this is us actually, we were out in the field planting a larger randomized block design of our S2 family so that we can perform some more complex genetic studies. And with that, I'd like to thank the current and former members of the SMART Lab that have contributed to this research. And I'll leave the rest of the time for any questions.
Thank you, Brendan, for that great talk. We got time for about 1 or 2 questions. So if you have a question, go ahead and raise your hand. Anybody have any questions for Brendan? All right. If not, join me in a round of applause, and thank you again.
Up next in our travel scholar is Matthew Davis from UC Davis. And he obtained his bachelor's in biology with a specialization in biotechnology and entomology. And he graduated in 2020 from the University of Florida. During his undergraduate studies, he worked with several blueberry projects, including optimizing vitro lighting conditions to improve transformation efficiency in blueberries. Currently, he's a first-year PhD student working on genetic adaptations to climate change. And today he'll be talking to us about his salt tolerance research. Thank you.
Hey, can everybody hear me okay? All right, awesome. First, thank you for the introduction. And also thank you guys for having me here today. I appreciate it. I also want to talk a little bit about salt tolerance in pistachio and the genetic control for this salt tolerance. So with that, let's get cracking. Oh, I need my, there we go. All right, so I'm going to start with a little bit of an introduction into salt tolerance and into this pistachio rootstock. So soil salinity is currently a threat to agriculture, and this threat is expected to be accelerated by the effects of climate change. So what we're seeing happen is as the water basins are depleted, they're leaving behind some solutes in the soil, so that makes the soil salty. But the big problem is that with these depleted water basins, there is only salty water available for farmers to use to then water their crops. And so that really compounds the soil salinity issue. And so if we look at this map right here, most of our pistachio production is in an area where there is high and critical overdraft of our water basins. So you can see salt is becoming a problem, yet pistachios are still growing there, right? So our pistachio rootstocks that we use are highly salt tolerant, and that makes them a good candidate for the future and for being a good nut crop. And so we're going to be specifically talking about rootstock today. The rootstock we're talking about is UCB1, and it's an interspecific cross, so that makes it pretty interesting. It is a cross between Pistachio atlantica female and a Pistachio integrima male. So if we really want to drill down to the genetic control of the salt tolerance, we have to do a couple experiments. So we started with mapping population of over 750 UCB1 individuals. And so my collaborator Latif actually grew out this population, and then he challenged them with 7 days of 100 millimolar sodium chloride solution, followed by 60 days of 150 millimolar sodium chloride solution, and another 60 days of 200 millimolar sodium chloride solution. So a lot of salt treatment. And then he went through and he actually counted the number of healthy leaves. So for example, this is what healthy leaves would look like, and this is what our unhealthy leaves would look like. After that, he collected leaf tissue samples and he sent those away for GBS. And he also collected samples to send for some further ionomic analysis. So we received about 29 different ion content information back, but we're going to be focusing pretty much exclusively on the chloride and sodium ions. So the first thing we wanted to check out was this healthy leaf phenotype that Latif had gone out and phenotyped for. But before I get into that, I do want to talk about a little bit what's going on over here on the x-axis. So we treated this population as a double haploid population because it's an interspecific cross. Our first 15 chromosomes are going to be Pistachio atlantica, and our second 15 chromosomes are going to be Pistachio integrima. And so that's the paternal parent on the right and the maternal parent over here on the left in blue. And when we looked at this healthy leaf phenotype, we found an association with a region on chromosome 13 in both the maternal genome and the paternal genome. Waited a little bit, got that chloride data back, and we found an association again on chromosome 13 in that maternal genome, and again on that paternal genome. And lastly, we get the sodium data back, and we're seeing an association in this same region. And so all of this taken together, we found an association with the number of healthy leaves, sodium content, and chloride content on chromosome 13 in the female genome, so right here on the left, and the male genome over here on the right. So now we wanted to look into further into the effects. So when plants inherited a high associated allele with sodium chloride for the maternal in the maternal genome and a high an allele associated with high sodium chloride in the leaf in the paternal genome, they had high sodium chloride in the leaf, right? So we see this nice easy trend. And obviously we're shooting in our program for this low because we want low sodium chloride in our leaves, or so we thought, and we'll talk about that in just a second. But after we started looking at this data, we wanted to come up with what potentially our mechanism for salt tolerance could be. So it could be an exclusion mechanism. We're just keeping that salt out of the plant entirely, and that would be ideal. It could also be some sequestration. Maybe it's taking up some of that salt and then it's just kind of stopping there. Not as ideal as exclusion because that could have some effects on growth, but also better than susceptible, right? Because then the plants don't die. And so to test this, we actually had some additional analyses done looking at the chloride content in the stem of these seedlings. And so we had them look at chloride content in the lower stem, the middle stem, and the upper stem. And when we looked at the chloride content in this lower stem, and then we looked at the plants that had inherited alleles associated with high leaf chloride content, they actually had lower stem chloride content. So the allele associated with high leaf chloride content had lower stem chloride content, and then the allele associated with low leaf chloride content had higher stem chloride content. That kind of threw our idea on its head. When we looked at the middle stem right here, we saw pretty much the same trend. And then when we looked at the upper stem, so the last third of the stem, that trend kind of disappears. So to make these trends way more obvious, I wanted to put them next to each other. So we named all of our alleles after the leaf chloride content because that was our first analysis that we did. So if you inherited a high leaf chloride content allele, high leaf chloride content, but low stem chloride content. So what we think might be happening now is our new proposed mechanism for sequestration is that this salt is actually being shuttled up through the plant and into the leaves. And to test that, of course, we had to do some field trials. So what we were doing out here is these seedlings were actually planted from that initial greenhouse experiment that we saw. We planted them out in a saline field. So this field has about 100 millimolars of sodium chloride in it. And then they were grafted like they would be for normal production. After about 3 years, the PI, Pat Brown, went out and took caliper measurements. So that's essentially telling us our tree diameter. And then when we did some QTL analysis for that, we found the same region in the paternal genome. So Pistachio and Tegarima on chromosome 13 seem to be associated with caliper. So this work is still ongoing, but I'm going to recap with where we're at now. So we have an allele associated with healthy leaves, lower chloride content, and lower sodium content in the stem. But that seems to be an in the leaf, but that seems to be an inverse relationship with the stems. So high sodium chloride in the leaf means low sodium chloride in the stem. I think this is a very good lesson for an early PhD student. Mechanisms are important and field trials are important. So we need to make sure that we know 100% what's going on. If we had made our selections out in the greenhouse, then we would have had a major issue because the plants that we thought were unhealthy were actually our healthy plants. And then I'm going to float this out as a little bit of food for thought. So pistachios are deciduous. So this is actually a pistachio and Tegarima, so our male parent. They're planted all over Davis as an ornamental crop, and they obviously drop their leaves. So maybe this system is not ideal, but might be okay. So we shuttle the salt up into the leaves, we drop those leaves at the end of fall, and then we have a chance to create new leaves and get rid of our salt content. So what's next for us? First thing that we are going to do is fine mapping. So we've actually already created a fine mapping population. We are currently in the process of extracting the DNA for that, and then hopefully finding our candidate genes. And then after that, I think it'd be really interesting to look into our mechanism of salt tolerance. So how are we shuttling this sodium chloride up from the stem into the leaves, and why is it not being sequestered in these other tissues like our root and our stem? And with that, I'd like to thank everybody in the Brown Lab, as well as the Baxter Lab at Washington University in St. Louis for the inomic analysis, and the Mitchell Lab at UC Davis for their UCB1 genome assembly. And I can take any questions that anybody might have. Thank you.
Thank you for that interesting presentation, Matthew. We'll take any questions from the crowd now. If you have a question, please raise your hand. Do we have them back here?
I have a question.
Yeah.
Now you're working with the rootstock.
Yes, we are.
So what happens when you graft it?
Yeah, that's a good question. So we've been out there in the field trials with the grafted plants for about 3 years. There's a lot of questions about yield data. There's going to be a lot of questions about sodium content in the leaves. So this was kind of a project that had been put on the back burner, and with all this data, and I came in and did the analysis. So I think that's something that we're going to be looking at this next season when the leaves drop, is that salt in the leaves. We did find a preliminary analysis, a result of some data that showed that what we thought was the good allele, so the low leaf chloride allele, had actually a lower caliper out in the field.
We got a question from the chat.
Reading this from the chat. Well done for a beautiful presentation. First, I joined about 10 minutes ago and you might have answered my question. What information do you have on chromosome 9, the peak of maternal genome for chloride content? This is the same for the tree diameter QTL figure?
Yeah, so maybe not, but chromosome 9 is super interesting. Okay, awesome. So there's a large, well, tree diameter was right there, I just passed it. So there's a large peak right here, very significant. We've seen this peak in chromosome 9 in multiple traits, and it seems to be associated with overall tree vigor. That is something that the Mitchell Lab is currently working on, as well as the Brown Lab. So there's, I think, some stuff to be coming out about that soon.
Another question in the back here.
Wonderful presentation, Matt. I got a quick question.
Yeah.
So you talked about exclusion being a better mechanism versus sucking it all up, essentially.
Yeah.
Is it possible to actually use the plant as some sort of mechanism to just soak up all the salt into the plant itself? So perhaps that will decrease the overall salt content in the soil for next year or later planting seasons.
Yeah, that's a good idea, and it's something we've been kind of talking about. It depends on yield data, but if we could potentially use it as a source of phytoremediation for the soil, getting rid of this sodium chloride, it would be super great, because then we can also produce pistachios on that land at the same time. But fortunately, it takes about 6 years to get any good yield data, and we're at about year 3, so lots of waiting.
Simple question. How long do pistachios grow in the field before they replace them as a?
Yeah, I think it's typically around 15 years, but there have been cases of trees that are in for about 30. So it's variable, unfortunately.
And did you say how many acres there are of?
So I didn't say how many acres, and I don't know off the top of my head the amount of acres. I do know that the US is now the #1 producer of pistachios in the world, and California produces about 98% of those pistachios.
Does the salt content being more in the leaves, does that also mean that it's more in the nuts as well?
Yeah, another good question. So I mean, that might be really cool, because then we don't have to salt the pistachios, right? But yeah, we're not 100% sure yet. We're still waiting for the yield data. I think it'll be super cool in 3 years, maybe nearing the end of my PhD. Hopefully we can get some information in there about that. But as of right now, we don't know.
Got time for a couple more questions. Any more from the crowd? Looks like we got another one in the chat, maybe.
This is a follow-up.
Mm-hmm.
They want to ask, is your data collected across several locations? If not, would you mind checking the variability across locations?
Yeah, so the data is collected at one location right now. So what was the second part of the question?
The variability across locations.
Oh, okay. So it's one location right now, but with this new analysis, there are plans to expand.
All right, any more questions for Matthew? All right, join me in a round of applause then.
Thank you, everyone.
Our last travel scholar speaker.
Hello, my name is Z, and it is my pleasure to introduce our last traveling speaker. His name is Sadeep, and he is a PhD candidate in the School of Plant, Soil, and Environmental Science from Louisiana State University. He has completed his master in South Korea and is now pursuing a PhD in the crop plant genetics lab under the supervision of Dr. Subaruti. And his PhD research is mainly focused on the abiotic stress tolerance in breeding and genetics. And the specialty is in salinity tolerance in rice breeding. And please give a round of applause for our next speaker.
Thank you for the nice introduction. Good afternoon and a warm welcome to my presentation. I'm Sandeep Sapagai. I'm from Louisiana State University, and I'm working on the abiotic stress tolerance rice breeding program. My PhD dissertation research work is focused on the salt tolerance breeding program. So I'm going to present some of my research approach to develop the salt tolerant rice varieties breeding lines in US rice cultivar. We all are familiar with the rice. This is a very common crop and it's the third most major crop in the world. And in USA, there are 6 large rice producing states. The Arkansas is the top most rice producing states, followed by California, Louisiana, Mississippi, Missouri, and Texas. And the rice consumption is increasing in the US as well these days. And the previous speaker, like Matthew, he discussed about the salt stress earlier. So approximately 11.25 million hectares of the land, it is affected by the salt stress, and 1/5 of the irrigated land, this is salt affected. And the 1.5 million hectares every year, it's becoming unsuitable for the agriculture production. So this figure, this shows the stage of salt stress in the rice. The rice, it is a little bit more tolerant in the germination stage, but it is susceptible in the seedling stage, panicle formation, and the flowering stage. And that means it is more susceptible in the seedling and reproductive stage than the germination stage. Seedling death occurs when the salt stress is more than around 10 EC, and we can see the significantly reductions while the salt stress is around 4 EC. So this figure is my own experiment. What we saw, you see the first one is the control seedling stage, and this one is the segregation, the 12 days after the salt stage. We gave 12 EC in the hydroponics and the 18 EC salt stage in the sand culture. And this figure shows the reproductive stage screening. This one is the control for the reproductive stage, and this one is the stress after the 8 EC after booting stage until the maturity. So this simply shows how the salt effect, stress effect in the seedling and reproductive stage. This is common for all the breeding approach, right? So I don't want to discuss about this one. This is a basic approach for develop the abiotic stress tolerance crop varieties. So till now, for the QTL mapping, there are several studies for the different stages of the salt stages in the rice. But the saltol QTL, and this is the mainly popular, and it has been cloned. The SKC QTL and the QSNC, those are the major QTLs identified for the salt tolerance. Since the salt tolerance, this is the salt stress is the abiotic stress, it is governed by the polygenic effect. So marker stress selection, this is easy to select the target QTLs, but it is more challenging due to the complex genetics of the abiotic stress. So we don't know the level of tolerance of introgressal lines developed by our varieties, cultivars, may or may not be good as a donor line. So basically, we have the three different approaches to develop the salt-tolerant breeding lines. First one is the development of the biparental populations. We selected the Louisiana variety senior, and we use the TCCP as a donor parent for the biparental population. And second one is the development of the multiparental populations. For the multiparental population, we use 2 or 3 donors in the US rice background. And third one is the pyramiding of the targeted QTLs associated with the salt tolerance traits from the multiple donors. So this was the population development, how we develop the populations. So you see the senior is the recurrent parent, and TCP the exotic donor parent. And we develop the BC3F4 population, and we screen those lines in the salt stress. This is our screening method. For the seedling stage, we grew the plant, the seeds, until the 14 days, and we gave stress after 14 days as plant. First, we gave 6 EC salt stress for 3 days for the adaptation in the hydroponics, and we increased the salt stress to 12 EC, and we gave continuously gave the 12 EC stress for 7 days in the hydroponics. Whereas in the sand culture, the absorption is really slow in the sand, so we increase the salt concentration up to 18 EC in the sand culture. And then after the visual scoring, we took the other measurements like root length, shoot length, and then sodium potassium content. And we also took the chlorophyll content just 4 or 5 days after giving stress. This is the Erie standard salt injury score table. So we gave 1 to 9 scale. 1 is the highly tolerant, whereas 3 is the tolerant, and 5 is the moderately tolerant, 7 is the susceptible, and 9 is the highly susceptible. Okay, this histogram simply shows the distribution of the lines and the parents. So those are the measurements like salt injury score and shoot length, root length, spar unit. This is the chlorophyll content, dry weight, sodium concentration, potassium concentration, and sodium potassium ratio. This simply shows the distribution of the lines and the parents. This is a QTL table. So for this biparental population, we screen for the different growth stages to identify the common QTLs, because there has been no any study for the identification of common QTLs in different stages using the same population. So here we want to identify the common QTL in all growth stages so that we can use that QTL for the fine mapping, and then we can use the same QTL for the salt tolerance. So interestingly, we found this is the rough table. So we found like a significant high. You can see the 23% phenotypic variance, and 20% and 60%. These are some of the large effect QTLs. This is in the seedling stage. And then we are interested in the germination stage also, because since it is relatively higher, like relatively more tolerant in the germination stage. So for this one, we gave the different concentrations, the stress from the 120 millimolar, 140, 160, 200 to the parents, and we selected some lines, and we found 200 millimolar. It has significantly difference in each stage or in terms of number of germinated seeds. So we selected 200 millimolar for a whole population for the germination screening. So this is the table, QTL table for the germination stage, some of the traits. So we measure like speed of germination, germination index, coefficient of velocity of germination, mean germination rate, and final germination. So interestingly, we found the speed of germination, germination index, QTL, and the shoot root length QTL, shoot root ratio QTL for the seedling stage is same QTL. Rest of the other QTLs are different. So this workflow, this simply shows how we did for the reproductive stage screening for this biparental population. So for this one, first we planted all the lines along with the parents, and we gave the stress at booting stage, and we gave continuously ATC salt stress until the maturity, and we took the reproductive stage associated traits. And this is how we measured, evaluated the plant-wise. And these are some of the lines, good lines we found based on the phenotypic evaluation. So this is the second approach. Here we develop the multi-parental population. Here you see the we use the Pokali, Hasawi, and FL478 as an exotic donor in the background of Jupiter, Mormenta, and Senior. And the first population where the Mormenta is used as the recurrent parent, and we gave the name MPS population. And second population is the CF DJ population. And this is the first, we screened all the populations in the sand culture, and this is how we found the segregation from the highly tolerant to susceptible. And then we selected 18 lines from each populations, and we again retested in the hydroponics. And you see the first panel, these are the control panels, and those are the F states. And this is the visual salt injury score, and we can see most of the lines, all the selected lines are around 3 to 5 salt injury score. And sodium and potassium ratio, this is the major trait for the salt stress tolerance in any crop. So here we measure the sodium potassium ratio, sodium potassium concentration, and sodium potassium ratio. And here we see the sodium potassium ratio is almost the near with the donor parent like Pokali and FL478. And this is the, so we did the clustering based on all the major morphological traits, and we found, interestingly, we found all the recurrent parent are clustered separately, and whereas all our selected tolerant lines are clustered with the donor parents, so that we can say these are phenotypically clustered with the grouped with the tolerant parents. And then we are interested to know how the genotypic, they are clustered with the similar with the genotypic genomic composition. And we roughly took the 100 polymorphic markers, and then we found here you can see all the, for genotypically, the selected lines were clustered with the recurrent parent, and the donors are separately. So as expected, we can see those lines are genetically with the recurrent parent and the phenotypically with the donor parent. So we can confirm these are the salt tolerant lines, and they have the salt tolerance gene composition. The third one is the pyramiding of the QTLs. Here we use the integration lines developed by the order studies from our group, and I use okay, this shows the, before that, this is the performance of the donors. You can see that Pokali, we have the Pokali, the parental donors, and Nona Bokra. And our recurrent, the US varieties are Jupiter, Senior, and CL151. These are our recurrent parents. And then those are the performance of our lines that I use for the to develop the pyramiding population. This GN100 and GN520 I use for the crossing, and CLPK 683 and CLPK 757 I use those lines. Those are tolerant. And this is the simply population development scheme. And this is interesting. This is how I develop the salt tolerant breeding lines. I cross those tolerant ILs, and I tested F3 lines, and then we follow the same salt screening procedure, and then we transfer the healthy, the tolerant same seedlings to the soil, and then some of them started dying, and some of them they resurvived, and we got some of the plant they resurvived, and they gave us seed, and now we have the F5 generation, and we are again screening. So our future plan is to do the genome sequencing and RNA-seq analysis using those pyramiding lines, the tolerant lines with the parents. And then then all the agronomic and the yield associated traits will be evaluated in the field for the future variety release. So our expected outcome, we have already got some of the tolerant breeding lines, and then we will identify the salt tolerant genes and superior alleles, and which will be the tool for market state breeding to develop the salt tolerant rice varieties. So I would like to thank my advisor, Dr. Subudhi, and LSU Greenhouse Facility, and this project is funded by USD and NIFA. And again, I would like to thank this Symposium entire team for this opportunity. If you have any questions, I will be happy to answer them. Thank you.
Thank you, Sandeep, for that seminar. We got about time for about one question from the crowd. Do we have any questions? All right. Maybe I got one right back there.
Yeah, very good presentations. In our slides, you showed the resistant versus susceptible are very clear-cut difference. And you are putting into 4 or 5 classes. So if you take class 3 and raise the progeny of class 3, do you get class 3? As a breeder, I will get resistant 1 and rest everything is susceptible, and then your analysis becomes very simple. Unless you have proof that 3 and 4 and 5 will breed true for 3, 4, and 5. Because depending upon the environment and so on and the background gene, the same major genes show different variability. So to make it simple, you just have classify into 2 classes, tolerant and susceptible. Thank you.
Well, thank you.
All right. Thank you guys for coming back after the break. Our next speaker is Dr. Thomas Lubberstedt. Dr. Lubberstedt is a professor and KJ Fryer Chair at the Department of Agronomy at Iowa State University. He also serves as the director of the R.F. Baker Plant Breeding Center, and then he is also the director for the Distant Master Program. He is the founder of the Double Half-Life Facility on campus, and he is also the faculty scholar of Plant Sciences Institute at Iowa State. His current research primarily focuses on the DH technology and its application, which resulted in many USDA as well as FFAR granted projects, and that the DH program serves to generate desirable germplasm for both research and commercial purposes. This year, he is also serving as the co-host for the upcoming 2022 NAPH meeting in August at Iowa State. And I know Dr. Lubberstedt since my undergrad. He's a really funny person. And one of the rumors going on is that he eats red onion in his office like an apple. So hopefully you guys can spread that rumor and embarrass him continually somewhere else.
Not true.
All right, there we go. So I'll hand off the floor to Dr. Lubberstedt.
Thank you. So first, I would like to thank the student organizers for inviting me. It's the first time at TAMU. So I really enjoyed yesterday evening and thought it was an interesting day today. So I learned a bit about haploids and dihaploids. So particularly interesting for me. I wanted to start out with ancient history as it was stated in this subject of an email that I received in 2010. And I got it from a person called Chase. And I knew that Sherrod Chase was actually doing research at Iowa State in the 1940s on double haploids. So I thought that's probably a grandchild that wanted to know more about what his or her grandfather did. It turned out it was Sherrod Chase himself. He was congratulating us for establishing this DH facility. And he said, yet I must admit some irony in that such a facility should have been set up at Iowa State some 65 years ago, when my pioneering haploid work at Ames opened the commercial possibilities of double haploids, etc. He then also asked about my research plans ultimately, and that he would send me a package of reprints. which arrived about 1 week later. He's really a pioneer in the DH world. He has made groundbreaking work. He established the first DH line during his time at Iowa State. He really paved the way to make it commercially available. The basic fundamentals of using a color marker for haploid selection, genome doubling, etc. There are many things in these 6 years that he was already working on and successful. He was short after in 2011 and then also in 2017 visiting us to look at our DH facility and have intense discussions with graduate students and others. It was really an event when he was arriving. He was even giving a seminar in 2017 with the help of his already retired daughter. He was 97 by that time. And when talking to the new dean at that time, Dan Robinson, that we tried to get him nominated for the World Food Prize and failed, he said, we need to give him a special prize. And that's what we did. In 2020, he received the Cults Award for Sustained Excellence. He was 99 years at that time. And that was actually the last of these Cortiva meetings before COVID came. It was the first week of March. 2 weeks later, everything was shut down. So I'm really happy to be back here in person about 2 years later. I think it's one of the first of these symposia that are in person again. And this was also 2020. We did have the chance to, with some safety regulations and paperwork, to have a nursery. And it was just before the first storm came through, mid-July. Later, we had the derecho, so it wasn't the best of summers. I will provide some background why double haploids are used. I will mostly focus on optimization of DH technology and a little bit on utilization of DH technology for novel breeding strategies. Why is it used? It was already mentioned, this speeding up aspect that you could get DH lines. Chase was calling them also monoploids, the haploids actually. And there's some confusion when you deal with tetraploids and thus dihaploids, but I will stick to the term doubled haploids, which is found in the literature, which means diploid genotypes that are homozygous and you get homogeneous lines out of them. And you can produce them in 2 generations as opposed to maybe 5 to 8 generations that you would need to produce a DH line or a fully inbred line by self-pollination. So you can affect the denominator of the genetic gain equation, but in addition, DHs also impact the numerator by increasing the genetic variance compared to F2-3 or other types of families, because all genetic variation is between lines and there's no more variation within lines. That also increases the heritability, so your trait evaluation is better using homogeneous lines with homozygous genotypes compared to segregating families. In maize, in vivo haploid induction is used. In vitro methods were tried, but they were too genotype specific. There are 2 possibilities for in vivo haploid induction. One is the paternal route, where you use the inducer as a female. It uses other genetics, the indeterminate gametophyte gene, which has been isolated. You would pollinate the inducer, and the only application today that I'm aware of is rapid conversion of normal lines into CMS lines. Assuming that you have CMS in your paternal inducer, you can rapidly convert a line into a CMS version. I will focus in the following on the maternal haploid induction, where the inducer is used as a male. And that's always confusing. It's called maternal haploid induction while using the inducer as a male. but that is because the haploids that are really the main purpose are coming from the donor. And today, roughly 10% haploids can be generated in that way in a typical cross. The remaining 90% are regular diploid cross offspring. This is the process. You start with your cross, then about 10% will be kernels with haploid embryos, and a color marker, the R and J gene from the anthocyanin pathway, is used to help identifying those haploids. There's this double fertilization event that somehow fails, as we already heard from Joann, and it leads to still viable kernels with triploid endosperm, but embryos that only have the female genome. And because the inducer or inducers today carry this color marker, you can identify those kernels by having a white embryo, as you can see here as an example, as opposed to 90% of the kernels that have this colored embryo. These kernels, the haploids, are selected, then they typically get treated, and in most cases with toxic colchicine, which helps to double the genome. Because you are doing that under controlled conditions and on seedlings, you can no longer sow the seed, but you have to transplant those seedlings, which is the least favorite part of the work. And you get out of this chimeric plants, because when you treat those seedlings, you already have multiple cells in the embryo. Some double the genome, others not. So you have haploid diploid cell lines side by side. And then you have to be lucky that you have diploid cell lines that lead to pollen formation and egg cell formation. So it's a numbers game. You get only about 10% haploids from that cross, and only about 10 to 20% make it then to become a DH line. So if you produce 1 ear with 100, 200 kernels, maybe you get 1 or 2 DH lines out of that. And so it's not really efficient, and anything that can help increasing the efficiency would be welcome and save costs. One should say that the major breeding programs are using this process or a variant of it. And most of the inbred lines produced in the major breeding programs are DH lines today. So it's hundreds of thousands of DH lines produced every year. So you can improve that process by creating better inducers, automate the visual and manual selection, or work on this step of the genome doubling, which in fact I think would be the most important and rewarding thing if you could get rid of a chemical treatment and put the selected seed directly back into the soil if there was a genetic mechanism that would enable that. There's a pedigree of inducers. So the first real inducer that we heard about was stock 6, developed by ETCO. It had about a 2% induction rate at that time. So companies were not too excited early on. So it took decades before it really was used routinely in companies. From there, inducers were developed in China, Eastern Europe, France, and the modern inducers, and it's not complete, there are more inducers today from CIMMYT and others, but they are exceeding 10% induction rates. And we had access to an inducer that was developed at University of Hohenheim by Professor Geiger, and we started developing our own inducers from there. And I will talk more about those in the following. The motivation for us to develop our own inducers were mostly driven by adaptation issues. I mentioned we have storms every summer almost. There's a severe storm coming through. The Hohenheim inducer that we had initially wasn't adapted to that. So the inducer was on the ground. It was still alive, but we had to crawl on the ground to collect the pollen. And we decided we don't want to do that every year. And so agronomic traits maybe sometimes forgotten, but they are really important to have in your inducer materials if you want to use it over time again. Then seed set was another problem. We had very limited seed set in this Hohenheim inducer under our conditions. And so that is another trait that we have been looking in. I will talk more about induction rate. Once you start developing your inducers, of course, induction rate, you try to increase it over time. And I will talk a little bit about other markers or traits that could be interesting. In particular, oil content is kind of the latest in inducer development, that you would like to develop high oil inducers to facilitate haploid selection using a machine based on the oil content in the offspring that you're producing. The heterozygous offspring that you're producing are expected to be about intermediate between the oil content of the inducer and the donor. The donors today, the elite germplasm has around 3% oil content. Inducers, high oil inducers are 10% and higher oil content. So the hope is that the heterozygous kernels or the kernels with heterozygous embryo show oil content in between, somewhere 6–7%, whereas the haploid embryos are still around 3–4%, and that is shown here. But it also shows you get 90% of the heterozygous fraction and only 10% of the haploid fraction. So even if you can separate it, there's still a tail of the diploid fraction that is overlapping and leading to false positives in the haploid fraction. So it's really about trying to drive the means of the diploid fraction and the haploid fraction as far apart as you possibly can. So we started developing our high oil inducer, again motivated by having something that is also adapted right away by using our inducer that we had meanwhile available at about 12% haploid induction rate. And we crossed it in collaboration with the Illinois group with their high oil inducer that is at 16% oil content that has been coming out of the long-term high oil selection experiments. And this is still ongoing work, so I will not show any more results. We wrestled with the question, should we have about 50% genome contributions, or should we have more from the high oil genotype or more from the inducer, because both are ultimately quantitative traits. And that's something we are looking into. At the same time, high oil doesn't necessarily mean that per cell that you have an increased oil content. It can simply mean that embryos are larger. And that is quite typical that with high oil, you also increase the embryo size, which also means that you can, instead of selecting for high oil, you could select for the embryo size to discriminate diploids and haploids. And that's what we are attempting. So this Q sorter, which is available at our plant introduction station, can sort based on images, but also based on NIRS. And we have another independent collaboration in the frame of a USDA project, SweetCup, with Mark Settles, Marcio Resende, and others, and Jeff Gustin, who was there in this meanwhile in Illinois, to try to make this SK single kernel NRS machine work for haploid sorting. This machine was built by Paul Armstrong, USDA in Kansas. And this is going on, so we are decently optimistic that this year at some point we can get it to work. And we have a first 1.0 version high oil inducer with 10% for induction rate oil content available at this time. Other traits, just an example, once we got going with the DH facility, we got in touch with popcorn breeders. And popcorn is hard to select using this R and J markers. So color selection is a problem based on the kernels. Then often in popcorn, you have the cross incompatibility mediated by the GA1 gene. And in order to be able to work with popcorn and develop popcorn DHs, we decided we need an alternative marker we developed and utilized, not developed, but utilized the PL1 gene, which leads to red coloration of seedling roots. So we can now select haploids based on root coloration. The white ones would be the haploids. And GA1 just means that if you have the large GA1 allele present in popcorn, then these genotypes cannot be pollinated, for example, by transgenic field corn. So it's a mechanism to protect popcorn to receive transgenes, for example. Nobody wants to chew on transgenes when chewing popcorn. And in order to ultimately be able to develop haploids and double haploids in popcorn, you need inducers that also carry this large GA1 allele. And that's what we did and have since been working with popcorn breeders. We heard about genes isolated, matrilineal or the other names on the one hand, and ZMDMP. I would say that matrilineal is probably the main gene here. And it has been shown that if you create this 4-base deletion that was found to be causative in other non-inducer genotypes, you can get induction rates in the single-digit range up to 10% perhaps. And you can create it in other species, in some other species at least. But it also means just based on matrilineal, you're not able to really go to high levels of induction rates. Also, matrilineal is already present in the current inducers, including stock 6. So the question that we were asking and are asking is, can we use the best inducers today and do what breeders do, cross good by good and hope for something better? We had access to this Hohenheim inducer, RWS, but also to PHI3, a Moldavian inducer. And the question was, do we observe transgression? And the answer is yes. So we did find lines, F4-5 families. This was done by Henrique Trentin during his PhD that exceeded 20%. Unfortunately, Prossera, who owned PHI3, is no longer in business. That obscured the ownership question, who owns PHI3 now? And so our IP department said, don't touch any of that materials any longer. So we had to abandon these materials and could still use it then for some studies, but for developing high inducing materials, we had to start over again ultimately. What I wanted to just emphasize is that determining haploid induction rates is fairly laborious. You are doing a lot of crosses for each family that you're evaluating. You try to pollinate a couple of donor plants. We always use the same donor genotype so that we can compare between families, but also across environments. The goal is to get about 1,000 kernels per entry, per plot. And then you have to select for haploids in each of those. So it's really laborious. And for that reason, we are thinking that genomic selection and trying that makes a lot of sense. When doing that, you create a training population and you can just use the same data to ask, do we find any signals using the GWAS approach that was done by Henrique using these old inducer data. And he did find a couple of hits, is still doing the final analysis. What I found interesting was that he found a strong hit on chromosome 1. That's where matrilineal is located. And so there's a possibility in these materials where matrilineal is fixed, that there's perhaps even something else located on chromosome 1 affecting haploid induction rate. We use these materials as well as materials that was published from University of Hohenheim about existing inducers, a GWAS study for initial genomic selection experiments. And by cross-validation, we could show that haploid induction rate actually has a fairly good. Prediction accuracy comparable to plant height. So what we are taking from this initial study is that it's promising to apply genomic selection for haploid induction rate. In addition, what is really benefiting modern breeding programs is the combination of double haploids and genomic selection. And again, ironically, because all of these inducer families are fixed for the red RNJ marker gene, we couldn't develop the H lines for our inducer families, our candidates, because the marker gene is fixed. There's no way to discriminate haploids from diploids using the RNJ color gene. But we knew that there's another gene, the C gene, also in the anthocyanin pathway, which is epistatically suppressing the R gene. And that is sometimes a nuisance. We deal with flint germplasm from Europe or Argentina. And sometimes when we work with that germplasm, we are not able to work with it because selection doesn't work. The red coloration is not expressed. So we were asking, can we actually use it in order to develop inducers that are carrying the C gene so that we can then suppress the R gene and select for haploids that are expressing the color. And the remaining, the 90% of heterozygous diploid genotypes would show no coloration. And the answer is yes, it did work. So we developed the C inducer and we created a bunch of inducer DH lines using that tool, and we'll now use it in a genomic selection experiment by the follow-up student, the student Yuru, who's following up Henrik in attempting to use genomic selection plus double haploids for inducer development. And he had his first training population evaluated last summer, and the next round, the validation is coming up this summer. So to summarize this part, we have various inducers developed along the way, something we call Midwest inducer in the 10 to 15% induction rate. I think with the training population, we enter the 15 to 20% range, and we know it's possible to exceed 20%. So that's the longer term goal. It's possible to derive hybrid inducers, which could be of interest in terms of isolations. We want to have more vigorous plants that are spreading pollen at larger quantities. We have this GA1 inducer and are working on another cross-incompatibility inducer, the C inducer. We work on the high oil as a byproduct of another project we developed, a GFP inducer, GFP for selection. And we are interested in developing a transformable inducer. The motivation here is based on the paper that was published in 2019 by the Syngenta group of Tim Kellyher. They showed that inducers can be used as a tool to do editing very efficiently. And the idea is if you can transform your inducer, you can add in the apparatus for Cas9 and the guide RNA. Then you will have your double fertilization. Assuming it's chromosome elimination that is happening, there will be a contact between the maternal and paternal genome for a certain number of cell divisions during which editing can take place. And then by chromosome elimination, the paternal genome is eliminated, and with it, the transgenes are removed, the mutation resides, and if you're then lucky to get doubling occurring, then in one step, you're getting doubled haploid lines that are mutated in the target gene without any trace of the transgenes. And you can do it in any background because you can pollinate in any background. And we have shown that in principle, it can be done. So we can use the baby boomer Wuschel procedure to transform our current inducer. But we think down the road, it would be preferable to have a transformable inducer. So I'm now moving on to the donor side. I talked about the male side, now the female side. And one is the response to induction, and then the genome doubling. We have observed that there's using the same inducer, just looking at the blue bars, there's quite a range of response to induction of haploid frequencies that you find. That means there's germplasm that is more or less recalcitrant of being induced, and there's other germplasm that is doing quite well. So there's also genetics on the donor side that you can exploit, but we have no evidence so far that any major genes are involved. We did some mapping, others did mapping, and we are not focusing on that because we believe breeders will inadvertently select for increased inducibility over time once they move to a DH program. We are more interested in the doubling side because, as mentioned, if we are able to do that, use a genetic mechanism, we can avoid going into the greenhouse, the treatment, transplant. We can directly go with the haploid seed into the field. And the other question is, could we even be so lucky to find a major QTL or more involved in the process, because that would facilitate conversion of lines into lines that can be easily doubled. So we had a panel of inbred lines that was induced so that we had haploid lines that we could then evaluate for inducibility, but also for spontaneous doubling. So we were just planting out these haploid lines, and a student that came from TAMU was playing an important role here, Gerald Lafuente. And we looked for either tassel scores, so presence of fertile tassels, or for viable pollen, just pollen shedding, but you could also use some staining methods. And we're looking at a panel of about 100 lines. And as expected, the majority was sterile. And that's what you typically find for haploids, that there's no male sterility. The focus was on male sterility because it has been shown that haploid female fertility, for whatever reasons, is typically exceeding 50% or so. So if you would pollinate haploid plants with pollen from diploid plants, you get some seed set. The limiting factor in producing DH lines is the male side, and that's why we focus on that. And Gerald, as mentioned, he was really lucky to find a few lines, in particular GF1. GF is for Gerald de la Fuente, and later we revealed the real name in a publication. But GF1 was now really the genotype that we want to look into more closely. So we did a QTL analysis, biparental. We decided to create F2-3 families first, which were then induced to be replicated trials. And we did find for anther emergence and pollen shedding, one region in the shaded area on chromosome 5 that was showing up in all cases and had phenotypic variance explained around 40%, which means genotypic variance explained around 50%. There are some other minor QTL that we detected, but we would say this is a major QTL. And we are going on with fine mapping efforts. I can only say it's the coldest cold spot that you could imagine in a genome. It's really a difficult region, chromosome 5, close to the centromere, but we tried anyway. Then the next question was, does it work in other backgrounds? And Anderson Berzignazzi was working on BS39 materials, which was a tropical population adapted to Iowa photoperiod conditions. He used that to derive lines in different ways for different types of questions we wanted to answer. So he used a sample of BS39 to do single seed descents. He developed doubled haploids using colchicine. we took a sample of BS39 and crossed it with GF1 and then did the same, created single-seed descents or double tuplets, but the double tuplets were developed without colchicine, just using spontaneous doubling. And those were genotyped, and we were contrasting the double tuplets that were developed with or without colchicine. And using a case control approach, we again confirmed that this chromosome 5 region showed up strongly. And if you looked at the genome pattern at the haplotypes, then the DH lines that were derived from a cross with GF1, they were almost fixed in this chromosome 5 region for the haplotype from GF1. Which brings up the next question, is there any yield drag associated with it? And so Anderson was doing test crosses of these materials. And when comparing the single seed descents after crossing with GF1 or the double tuplets after crossing with GF1, we did not see a significant difference between those groups of about 100 lines in test cross versions for grain yield in this background. And also the range of variation was about the same. So at least in this background, there didn't seem to be a dramatic negative side effect. This is summarizing the current status. There's also other groups, CIMMYT, CAU, Beijing, working on this doubling trait. To my knowledge, this QTL on chromosome 5 is the only one that is really a major QTL at this time. There may be another one on chromosome 6 identified by the CAU group, and there's a couple of other regions and candidates. And while we focus on this chromosome 5 QTL and also screen for additional germplasm to find maybe additional donors for major QTL, we also are interested to look into candidate genes. And we are in an FFAR project using information out of these maze mapping studies to utilize the Arabidopsis DH system that Joann was already mentioning based on SENH3 to order mutants in candidate genes that reside in these regions that may have something to do with doubling, plus looking what are these genes doing and so on. So the goal is to screen 60 candidates in this Arabidopsis system. The nice thing is once you create haploids of Columbia, which is the background in many cases, there's a certain level of haploid fertility still present in Columbia. So that means we can look for both a decrease but also an increase in haploid fertility if we compare the performance of perfectly isogenic mutants at the haploid level. We are also working with engineers here to help with the phenotyping. And so far, it's not only 3, but a slightly larger number of mutants that seems to have an effect on haploid fertility. We did find mutants where it's going down, but we also did find 2 where it's actually going up. And so we hope that this work can be published later this year. This is the summer of 2021. So it's leading over to how can we now use this doubling. We are trying to really use it in this BS39 germplasm to see can we, without colchicine treatments, develop DH lines and do second cycle, third cycle breeding, etc. And we did get funding from the organic program within USDA for that. These are kind of second cycle haploids, and the green labels indicate they have been fertile without colchicine treatment. We are interested in the question whether we could use those fertile haploids for an accelerated breeding scheme where we would utilize the 2-generation scheme. We would, on the one hand, induce heterozygous genotypes and produce haploids which are fertile. That means there should be a chance that we can already cross at the haploid level. We don't have to wait necessarily until we have DH lines and evaluated them, but using genomic selection, perhaps we can just have a rapid 2-generation cycle. We can maximize the genetic variation in our breeding population by looking at gametes ultimately, which I would see as an advantage. We would take a certain fraction of genomically selected haplets as a male. Whatever is used as a male can also be self-pollinated. That means inbred lines could be developed and test crosses conducted to update the genomic selection model. And we would use another fraction of those haplets as female. And we could try to optimize what should be crossed with what and go back to produce heterozygous genotypes, which would be induced again, and then go to the next round of haploid crosses. So this is something that we are attempting, and we are not sure now how well it will work. We will see. But it also brings up a theme that I heard a few times with the potato talk, but also from Joann, that relates to the vigor of these haploid plants and the ability to produce seed on these haploids. So the female fertility and the seed set on haploid plants that we have ignored so far, because we said the male side is really important if you want to have DH lines. But if you want to use this accelerated breeding scheme, it will also be important that we have haploid plants that can set enough seed to move to the next generation. And what I wanted to say is the dogma is haploids are always worse than the inbred diploid. That's kind of what we see in the field and what we think is the case. But when you look at algae, there are so-called isomorphic biphasic forms where haploids and diploids are indistinguishable. And in some environments, you can even have something called haploid dominance. So you can have even more haploids there than diploids. And so I would question the dogma a little bit whether, generally speaking, haploids have to be always inferior. It could be driven by the fact that haploidy usually is connected with sterility. So there's no way to select for increased vigor because it's like a dead end route. Once you are haploid, you cannot contribute to the next generation. But if you now have this haploid male fertility, we can try if it's possible to select actually for more vigorous haploid plants. And so that's kind of what I'm now getting curious about and excited about. That's something that I would like to take on and develop as a new project to study something I would like to call haploid frailty. It's kind of similar to better parent heterosis. You would compare the better of the parents with the performance of the hybrid. And then the difference is this better parent heterosis. In this case, of course, you have the inbred, and typically the haploid would be a little inferior, and this difference would be the haploid frailty. And I'm curious if there's genetic variation. If you look for this haploid frailty for a range of perfect isogenic pairs, because they are perfectly isogenic, they only differ by having one or two of the same genome. If there's genetic variation for that, perhaps there are cases where the haploid is fairly similar to the inbred. Maybe there are cases where it's very different. And then even within a given comparison, you can ask if how that differs for different traits and how it looks at the expression level, etc. And that's all I wanted to talk about. So the conclusions, it took decades for sure for the DH technology to become routine. I think first in the 1990s, 2000, it really became routine in the major breeding programs. The main limitation was availability of effective haploid inducers, and I think that has been overcome, including the selection portion. I think that is not really a problem any longer or will soon be resolved. I think that the automated selection is ongoing, or in some cases, it's already implemented in breeding programs. I think the next frontier will be this doubling. I hope it can be used, and more research needs to be done. We are working with different partners to test it in sweet corn and different other backgrounds, while also looking for additional sources of this spontaneous hybrid genome doubling. But I hope that can also help breeding programs. And there are novel applications such as hybrid induction editing, which I feel is a very exciting tool. Having this fertility in hybrids can also open up new breeding schemes ultimately. Before closing, I wanted to repeat what C has been saying, that we will be hosting the 2022 NAPB meeting, and we believe and hope very much it will be an in-person meeting after 2 years of online meetings. So please take a note, it will be August 8 to 11. The registration will be sent out, I think, by beginning of April. Because we wanted to wait as long as possible to be confident with the COVID situation that we can handle it in person. And with that, I want to thank persons in my group, in particular Uschi Frey, who's managing the DH facility. There's a bunch of PhD students right now. Tyler is another TAMU product. We get funding from USDA, there are organic projects, the Sweet Cup project and the FFAR project, and we work with a couple of breeding companies. Thank you.
Thank you for that great talk, Dr. Lubberstedt. We'll open it up for questions now, crowd. So if you have a question.
Thank you. That was very interesting. I might have missed it. What was the motivation behind developing inducer line that has high oil?
Oh, that was, okay, can I? So the point is that you don't want to do the manual visual selection. The standard is that you're still using the color marker and do it by hand ultimately. And what you would like to do is to dump your seed into a machine and get out the hybrids. I think that's. Oh, okay. So you need a calibration for oil content, or let me go back now, or for the too many slides. So you would use either the oil content. And so the idea is you have an inducer that has an oil content of, let's say, 10%, 15%, and your donor typically has around 3%. And so if you create your typical cross, 10% hybrids and 90% seed you don't want, the seed you don't want has a diploid embryo. Oil is usually in the embryo, and hopefully the oil content is somewhere in the intermediate between what you have in the inducer and in the donor, so around 6–7% perhaps. And if you have a calibration that can separate and classify, put your kernels into 2 pots based on this classification, then you can kind of automate the process. So in this case, for the SK NIRS, which was built by Paul Armstrong, it's tumbling down here, and while it's tumbling down, an NRS spectra is captured, and you need the calibration before, which is taken with NMR. And if that is a good calibration, then you may be able to separate. And at the bottom, while tumbling down, and when it reaches the bottom, then it's going one of 2 directions. And it's similar here. So the speed, I should say, is in the range of 1 to multiple kernels per second. So it's really a fairly rapid process. I heard that a company is already using the Q sorter in their program, so it's not unrealistic that it will be more broadly available. So I'm pretty optimistic that it will work. Also, the Chinese and Hohenheim group, I think the Chinese group, they built a piece of equipment, CAU, based on NMR, which is, I think, commercially available.
Another question ahead.
I have a similar question about where you flip the color marker. So basically the color is now in the embryo instead of the endosperm. Can you do that through a sorter?
But we for doing IG1, and it's also publicly available. I'm not sure if somebody attempted it. If you think of it, if you have an inducer as a female, the limit that you have is the seed that you can produce on that inducer, which is maybe 200 kernels, and then you have a certain percentage will be haploid. If you have a male inducer plant, you can pollinate many, many donor plants. So you can produce much more seed and then get more haploid gain if you want in that direction.
We do have a question from the chat. The significant marker on chromosome 8 for USDA OREI ACEs, is that due to error in the reading the marker chip or a random effect? And aside from the oil content, what other qualitative advantage does the double haploid maize seed have?
So for the chromosome 8, we haven't really looked into that more, so we can't really tell. The main question that we tried to answer in that paper was, do we find the chromosome 5 region or not? It was a case control study, and that was all we were interested in. And the other question was?
Yes, the second one was, aside from the oil content, what other qualitative advantage does the double haploid maize seed have?
Yeah, I mean, you would use the oil content only to weed out, if you want, the kernels that you don't want. So the kernels that you're selecting would be carrying the regular genome of the donor. There would not be a trace of the oil inducer in most cases, I should say. I mean, there's a few cases reported that something from the inducer is coming through. So there can be a low percentage of haploids or DH lines that get a segment from the inducer that has been reported, but the majority would not get any genetics from the inducer.
Yes, they followed up and said, sorry, they meant quantitative advantage, quantitative versus
Yeah.
qualitative.
Yeah, but would be the same answer, I think. I mean, the main difference here is that you use a quantitative trait for classification, so you have to deal with these distributions as opposed to having the yes/no type of markers like red color, no red color.
So Tom, did you say that chromosome doubling, that you lose a lot of plants in that process? In other words, you create the haploid, and then in the doubling process, you only get about 10%.
Right.
So is there ways to improve on that doubling process? 'Cause that's
Sure.
a lot of loss. That's a 90% loss.
That's what I say. So it's really a numbers game.
Yeah.
And so even after colchicine treatment, and also there's some genotype dependency, but what we find it's 10 to 20% that make it through. And that's simply because you hit the meristem that then gives rise to organs and so on when already multiple cells are there. There are other procedures that have been described, N2O, for example. They reported higher rates of doubling, but they use also special chambers. So I think the numbers that you can run through are fairly, that would limit the process. And so the doubling, the spontaneous doubling that we observed was in the 70% range of this GF1. But of course, when we integrate it into other backgrounds, it's not as high. But I think we can get close to levels that we would get with colchicine. And I didn't show, we did have some collaborative work and compared the doubling being present or not present and using colchicine or not using colchicine. And at least for some genotypes, you can have an added benefit, have that doubling region present, but could still boost the doubling frequency further by adding colchicine.
You talked about combining gene editing with haploid induction. Is that in theory or has actually been tried?
It has been shown, has been demonstrated. And I should extend, has not only been done and shown in maize by this Syngenta group, but maize has also been used as inducer in wheat. So you can use the same in wheat. You could use the same maize inducer and apply it on wheat, perhaps other cereals. And I know another group in Gardersleben, Germany, they have been active. And I think they showed some nice example of converting 2-row to 6-row cereals or something like that. Very dramatic changes using this process.
And at which time you do the gene editing? Do you do gene editing on the inducer and then cross to develop the the haploids, or at which time you have the?
So you have to transform the inducer to carry the Cas and guide RNA, and then you would pollinate a donor.
Okay.
And has been this debate, and it's not resolved. Is it pathogenesis or is it chromosome elimination that leads to the formation of haploid embryos? So it could be that it's just the sperm is only inducing the egg cell to get going and develop an embryo, but there's no connection between genomes. And the other one is that you have both genomes in contact, and then you have a few cell cycles, and then somehow the male genome is kicked out again. And I think the group in Syngenta showed, and also the evidence coming from that once in a while you do find haplotypes blocks from the inducer in haploids or DH lines, that at least in some cases chromosome elimination happens. And if that is true, then there's some limited time where both genomes are together and editing can happen. And then the nice thing, as mentioned, is that the induction process takes care of the male genome and the transgene sitting on it, while you have then hopefully some plants with a mutation and the doubling and get your DH plant in one step. And I think the elegant thing is that often you're dealing with recalcitrant genotypes, etc. As long as you have a transformable inducer or a procedure to transform an inducer, you can apply it to any germplasm. You can pollinate whatever you want.
All right, with that, I think we'll wrap up questions for Dr. Lubberstedt. To join me again, another round of applause. If you have further questions for Dr. Lubberstedt, you can catch him after the symposium at the social. So with that, we're going to get ready for our last speaker. And Fabian's going to come on up for the introduction.
Our final speaker for the day is from our home team. This is assistant professor in the Department of Horticultural Sciences here at Texas A&M. Works on rose genetics and breeding with the goal of developing, testing, and releasing improved varieties of roses with regional and national adaptation, as well as conducting research on the use of genomics-based tools for rose variety development. So please welcome Dr. Oscar Riera-Lizarazu.
Thank you. All right, it's working well. All right, thanks for the introduction and thanks for the organizers for inviting me to speak to you today. I'm a relatively newcomer to Texas A&M, and likewise, I'm a newcomer to the world of roses. It's been exciting and interesting. I learn something every day. And what I'm going to try to do today is share with you some of the activities that we have undertaken to bring more genomics tool use to rose breeding, particularly the polyploid versions of roses, autotetraploids. And oftentimes, other workers that have worked with autotetraploid roses have taken the route of least resistance, working with diploids rather than tetraploids, or diplodizing tetraploid data sets. Well, nowadays, with the development of tools and the computational, I guess, capabilities, have created a different landscape where you no longer have to shy away from autotetraploids, even though they're still genetically complex. So we've got now the tools to do some of the things that we can do with diploids quite well. So I'm going to give you some examples of how we're going about that. But before that, I have to always give you at least a brief introduction of roses if you're not acquainted with them. Roses are native to the Northern Hemisphere. They were domesticated in, I guess, two parts in the northern latitudes, Asia and the Mediterranean, Europe and Middle Eastern region. Materials in the eastern side were mostly diploids with recurrent flowering. The material that was used in the western side were mostly tetraploids that were once flowering. And I guess through exchanges, these gene pools came together, and over time, the recurrent flowering phenotype was introgressed into tetraploids, which now gave rise to all of the tetraploids that we have today that are recurrent flowering. And by recurrent flowering is that they continuously produce shoots that flower throughout the whole season. Once flowering, it's a genotype that flowers once in the spring, perhaps, or maybe another time in the fall, but that's about it. Most commercial varieties are recurrent flowering. There are about 100 to 200 species in the genus, and 8 to 10 have been extensively used for breeding. So if you think about roses, it's really a complex of diploids, triploids, and tetraploids, and all of them are interspecific hybrids in many respects. So that's why the word hybrid is big in my slide, because that's as hybrids as we go with roses, because the way we breed them, we basically intercross F1s to F1s to F1s to F1s to F1s to F1s to F1s. It's hybrid by hybrid by hybrid by hybrid by hybrid by hybrid, etc. So they're highly heterozygous in addition to being F1 hybrids. In most cases, not in every case, but in most cases. We use them for the cut flower industry, roses. And you guys experienced Valentine's recently. So you're producing a lot of roses. Those are not the roses we work on. We work on landscape roses or garden roses that we said these would be materials that are planted in your garden rather than ones that you find in a flower shop, for example. Roses are beautiful, but they're also economically important. And as you can see, in the US, we produce about 25 million roses any given year. That generates about $168 million in sales. And some economists have estimated that direct impact of the rose industry in our economy is at least $772 million, if not higher. Okay, so roses are still very important economically, in addition to being, I guess, beautiful from an ornamental point of view. If you're from here, you understand this slide. If you're not from here, there's a couple of things that I want to illustrate with this particular slide, is that we don't live or work in an environment that is really good for roses. If we think that the optimal temperature range for roses is between 15 and 25 degrees, you see that in the growing season, we get up a lot higher than that in maxima and minima that are in the red and blue graphs there. And the, I guess, brown bars are the number of days that the minimum temperature is above 17 degrees C and the maximum temperature is above 30. So you can see that the entire growing season, and particularly the summer, are just completely stressful for roses. Okay, so that's one thing. Abiotic stress is real for roses, and it's one of the major challenges that the breeding program needs to address. The next graph is basically the same thing, maxima, minima with respect to temperature, and then in the blue bars, we've got precipitation. So we see that the falls and the springs are very wet, but even throughout the summer, there's quite a bit of rain. So it's also a very humid climate. Consequently, we get a lot of diseases that we don't get in more northern climates that affect roses, particularly fungal diseases. Okay, so this is an environment where biotic and abiotic stresses are very, very important. Therefore, a major focus of the breeding program. All right, some history about the program. The program was built by Dave Byrne, who's present with us here today. And the program was built on an interesting turn of events. There was a Texas A&M mathematician known as Robert Basey, who was also a hobby breeder. And in 1991, he decided to establish an endowment for rose research and contribute, of course, dollars and also germplasm. And that was the beginning of the rose breeding program here at Texas A&M. And Dr. Basey asked Dr. Byrne to take over and lead that program. Subsequently, in 2008, Ralph Moore from California donated his entire collection of genetic materials to the breeding program here at Texas A&M, which then became sort of the backbones with respect to germplasm upon which the program was built. Of course, Dr. Bin's been bringing in material from other places and introgressing species materials into his breeding program. But again, what we have today in the program really dates back to these major contributions of germplasm upon which all this other breeding research has been built. All right. This is just to give you an idea of the level of diversity. Yeah, we'll see in the germplasm donations that were made to the program. Ralph Moore utilized a number of species, and so did Basey. And you see that there's quite a bit of diversity from related species in the germplasm donations that the program has received. Therefore, the diversity is quite high in the program as well. All right, so the rose breeding program, again, the major objective, as was stated in the introduction, is to develop material that is adapted to our region here or any other humid subtropical climate. The principal diseases are two with respect to fungal diseases. We've got black spot and cereal leaf spot. And there is a very deleterious and important new viral disease that we call the rose rosette Emara virus, which is transmitted by microscopic mites. And this is the greatest threat that the US rose industry has today. You can't find this disease outside of the US at this moment. And we're very much focused on that disease over any other, on top of the ones that are, of course, of relevance to our region, which will be the two fungal diseases, black spot and Cercospora leaf spot. And so you will see that we put a lot of effort in that particular new viral disease that has to be controlled. And as you will see later, finding sources of resistance is still a challenge. On top of that, of course, flower productivity, particularly during the summer months, is another constraint that we have to address. So those are the four core traits that we normally look at, in addition to, of course, defoliation that can be also a consequence of disease and also a consequence of heat stress. Okay. This is just a little cartoon to give you an idea of what the scope of the program is with respect to materials that could be turned over any given year. And of course, this is just an example. There are years we have more material, there will be years we have less. But in essence, it has those particular phases. First 2 years is generating material that we can grow in the greenhouse for screening. During that time, and the greenhouse conditions will screen for things that we can screen there. For example, mildew resistance, recurrent flowering, flower color, morphology, growth habit, and that sort of thing. And in that example, we were selecting for recurrent flowering with Dave. And you can see we started with hundreds of seedlings from a particular cross, ended up with a very few that were actually recurrent flowering. And that's a trait you can pick up even at the seedling stage. Once those plants are potted, grown a little bit more, and moved to the field, then you start doing a lot of field assessments. And that was the years 3, 4, and 5. We're looking at traits that have to do disease resistance and other adaptation traits, and then ornamental quality. Then after that, if something looks promising, we give it to maybe a business partner who will then test the material to see if they will be willing to commercialize it. So there's a minimum of 7 years to produce material that could be commercial, but a breeding cycle is more close to 10 years because there's always an extra year that you have to do this, an extra year you have to do that. You got to propagate the material, etc. So that's just to give you a little bit of an idea of the length of the breeding cycle. And those arrows just point places where you could probably start identifying materials that you want to incorporate in a breeding block for crossing and additional improvement. Pretty typical of a lot of programs of this kind. So one of the things that as I said before, we're interested in is, of course, how do we integrate tools into the program, particularly genomic tools. And this is the simple approach that we're taking, is try to identify, map, and characterize genes or QTLs that affect traits of interest. Try to get that information and understanding applied into plant breeding context, and then try to accelerate some facet of breeding along the way. And as everybody is doing, we're also interested in exploring genomic prediction approaches to maybe get to the same goal, depending on the complexity of the traits that we have to modify or manipulate. This is a slide that I could not show you 2 years ago, but I can show it today because a lot of things have happened with respect to tool development in polyploids. So we've got 2 major genotyping platforms. We've got an axiom array that's extremely expensive, because if you want to say, well, how many public rose breeding programs use the axiom array, we would be the only ones that use it. So, using the axiom array requires a business arrangement, and the business needs to make money, and the genotyping costs for us are extremely high. So we wanted to move away from the axiom array towards a reduced representation type sequencing approach, or GBS as this is called more frequently. And we had done it in roses, or at least they did it in roses before I came here, but we had not really developed the approach for tetraploid roses because the complexity of autopolyploidy needs to be taken into account. And not until recently, with a collaboration with Trish Klein, we were able to actually make it work for us as well. So we have now the possibility to do that type of genotyping. And after that, you've got a number of little tools for dosage calling, haplotyping, mapping, QTL analysis, visualization tools that I'll talk a little bit more about, and of course, even genomic prediction tools that handle autopolyploids as autopolyploids. The mapping software treats autopolyploids as autopolyploids, and so does all these QTL analysis tools, and also a GWAS tool that does the same. So we can do all of the basic things with respect to gene discovery, if you will, in autotetraploids now, okay? And I'll give you some examples of how and who is behind some of these things in our group as well. So if you work with other tetraploids and you feel threatened by them, but you like to work with them, you can try to find our website, thepolyploids.org. It's a project from the USDA SRI program. And again, we've got connections to the developers, the tools, and know-how information. We perform workshops yearly on January. Our second workshop just happened. We had one last year. All of the videos that are both theory describing and also video showing on how to use the software can be found there. So if you want to get broken into autopolyploid genetic analysis, that's a good place to start. And you can also talk to us if you have some questions. Okay, so that's a plugin for tools for polyploids project. All right, I'm going to just give you some, show you and share with you some case studies on how we've been applying the tools. And again, we work with diploids just as much as we work with tetraploids. And in this particular case, we're looking at black spot disease as well as cereal cercospora leaf spot and rose rosette disease. And what I have here are very interesting materials. What we have are two interrelated diploid populations that were used for this example, and one interrelated population in tetraploids. So we have two interrelated families in diploids and one interrelated family in tetraploids. And by interrelated, I mean that the populations share a parent, okay? And this material was phenotype for black spot, cereal leaf spot, rose rosette disease in these locations. Rose rosette disease, we don't really have it in College Station. It's in the state, but not in our region yet. So all of the tests are done in Crosby, Tennessee by Mark Wyndham, who's our collaborator there. In College Station, we look at, again, black spot and cercospora leaf spot primarily, okay? And these were the years that the data was taken for the various populations. Some characteristics, the interrelated populations were GBS before my arrival, and so was the tetraploid population, and that was done with the Axiom array. And this is some of the software that was used for this particular case. And again, the interrelated population 1, that I call it for diploids here, has 9 parents, or I mean, sorry, it represents 9 populations, and the interrelated population 2 is about 8 populations. And again, in the case of the tetraploids, it's 2, okay? And we've got flex QTL that can handle interrelated populations in the case of diploids. And we use dia QTL for interrelated work, or QTL poly if we want to map each population individually. The interrelated populations of diploids have to be analyzed with flex QTL because the population size is not very large to be mapped or analyzed individually. And that's what we're using. So this is black spot, again, worldwide in importance. There are 13 characterized races, and there's about 4 race-specific genes that have been identified, and they're called RDR1, RDR2, RDR3, and RDR4. RDR1 has been cloned. RDR3 and RDR4 have been mapped to a genome location, and RDR2 hasn't really been worked on. Besides that, there's a number of QTL for partial resistance that we have mapped and other folks have mapped as well. So what we do is basically do field-based assessments. We don't work with race-specific inoculations, but with whatever's available in the field, natural inoculation, and we do monthly assessments of disease incidence, and we use a 0 to 9 score, 0 meaning no lesions on the canopy, and 9, over 90% of the canopy is covered with lesions, okay? And that's the normal scale that the program has been used historically, and we continue to use it now. So this is the summary of the QTL that we've identified so far. And the summary is we've got about 13 QTL in all linkage groups except linkage group 4. The red triangles map the location of 3 major genes, RDR1, RDR4, and RDR3. The blue arrowheads mark what they call meta QTL. So somebody did a meta-analysis of all the QTL for black spot, and those will be the locations of so-called meta QTLs. And then the rest of the bars show the QTLs that we have identified. Blue circles more show where QTL from multiple populations have detected that region. So there is some overlap with published QTL or major genes, but we're also discovering new ones. And a number of them. And then both diploid and tetraploid populations. Cercospora. I'm going to give you another case study for that one too. It's a disease that's important in Southeast USA. It's not a disease we see north of here, so it's more relevant to our region than others, but it's an important one. material needs to be resistant to that disease if it's supposed to be commercially successful. There are no known races. We don't know about resistance genes. Nobody really has studied except our group. So we will be the first ones to start identifying QTL for resistance to this particular disease. And again, we perform monthly assessments under field conditions as I described before for black spot. And so far, this is what we've identified looking at diploids and tetraploids, about 18 QTLs in all linkage groups. And again, there are places of co-localization of QTL. We've got the purple type dot, I guess, co-localization of QTL from tetraploid populations. The blue ones will be from diploid populations, and you see the spread of QTL elsewhere. And again, all of these analyses are preliminary, and we're starting to finish up some of the analysis to try to publish all of this and to verify and validate some of the findings that we're making. Finally, rose-rosette disease. It's caused by the rose-rosette Emara virus, and it's transmitted by a microscopic mite, Phyllocopterus fructiferus. And then the rose-rosette disease name comes from the spectrum of phenotypes that the disease produces. There's the formation of a rosette, and that would be a rosette. It's a very abnormal growth that you see versus normal in that particular plant. It has a strap leaves, stem thickening, hyperthorniness, and eventually the plant dies because of this abnormal development and so forth. So it's very deadly and very ugly, which is another reason why it's undesirable. As I said before, the field assessments are done by Mark Wyndham and Sarah Collins at the University of Tennessee. And the approach they use is taking material that is showing symptoms from susceptible roses, collecting tissue showing rosettes that also carry the airfield mite, that is the vector for the virus, and then pieces of that material is then clipped on material that's to be tested. And there is natural, I guess, infestation of those roses with rose-rosette disease from mites that are endemic, but it is augmented and supplemented by clipping tissue showing symptoms as well. So that's the procedure that is used for the field-based screening in Tennessee. Labor intensive, but that's the way it's done to guarantee, I guess, symptom expression. This is just an old table of a variety trial and survey that Dave Byrne and other co-workers had looked at. What is important here is to see that the majority of the rose cultivars, about 1,200 tested, were susceptible, and very few individuals show varying levels of partial resistance to perhaps immunity. And immunity, usually, we're talking about rose species or rose species hybrids, not really well adapted or commercial material. So there's quite a bit of work to go from here to try to move some of that resistance into cultivated types. So some of the work that we did for mapping involved materials showing some level of partial resistance, not immunity by any means. No silver bullet for rose-rosette disease yet. So on the field conditions, this is the severity scores that have been used. It's based on the number of rosettes, but also Mark and others have been collecting tissue and performing quantitative RT-PCR to detect the virus in tissues. And the CT values that the procedure reports is used as a proxy for titer. So we use that to also as a trait for susceptibility or resistance measures in addition to severity to rose-rosette. So this is done every spring and fall, and they've been doing it for 2 to 3 years. And that's what we're trying to summarize here. So when we take that data, we analyze it, we detect a major QTL in chromosome 5, another one on 6, and then some secondary ones in 1, 2, and 3. The 1 on 3, we see colocalization of QTL peaks in both diploids and tetraploids, which is very interesting. And it's the same case for the 1 on chromosome 6. Whether they are the same source or different sources, it's interesting that we see it in the diploids and the tetraploids. If they're different, it's a good way to start combining them into one, maybe one genotype with more and higher levels of resistance. So 5 QTL, all except linkage groups 4 and 7. This is just a summary then, 13 QTLs for black spot, 18 QTLs for Cercospora, and 5 QTLs for RRD. What do we do with so many QTLs? All right, so one of the things that we decided to do, and I'm going to show to you how I'm using some of that data to start, moving that information to more of a plant breeding context, is that I decided to use a tool called DIA-QTL that works with interconnected populations. And the example here is with tetraploid roses, but the same tool can be used with highly heterozygous diploid populations too, if they're interrelated or interconnected. But the example here is tetraploid because that is the tough one that we wanted to crack, or the nut that we wanted to crack. We use polyorigin, and what polyorigin does is it actually constructs probabilistic haplotypes for the parents and the progeny, meaning that it deduces the phased haplotypes of every parent and every progeny in those interconnected populations. And then that's what we feed the QTL mapping software to identify chromosomal regions that are associated with a particular trait. And if you're working with other tetraploids, you want to know where your QTL is, not the chromosome only, not the region only, but which homologue of all 4 is the one that actually has the QTL, because that's the one you want to use to move it forward. In diploids from homozygous parents, that's obvious. You know where it came from if both parents were homozygous, right? But this is the case of highly heterozygous parents. You need to know what the homologue is. You've got to deduce that. So that's a little level of complexity there. But we've got the tools to do that, which is great. So this is one example. And I'll walk you through a little bit so you understand some of the things that I show you. So we got 2 QTL, 103, 105, black spot. And 105 is huge because this is actually a major gene, RDR4. So it's a gene of large effect, and then we've got one of minor effect. Below, we've got what are called effect size plots. And what you see there is each bar corresponds to each homologue of that parent of that population. So you can actually see which homologue contributes to the trait significantly in both of these 2 QTL peaks. And in this case, migro contributes a favorable allele in chromosome 3, or homologue 3, I should say, of the same chromosome, in this case, chromosome 3. And then the QTL in chromosome 5, the favorable allele comes from bright eyes, homologue 3 of chromosome 5. So that is good to know. One favorable allele comes from one parent, a particular chromosome, and the other chromosome from the other parent. Then you can ask the software to tell you, well, what about the population? Who has what? So you can ask, well, how many of these progenies have inherited the QTL from migro? 85. How many inherited the QTL from 5? 166. And how many inherited both? 42. But they all pick up exactly the ones that have this and that homologue with the QTL. Okay? And that we could not do before that easily in autotetraploids, but now with this sort of software that breaks things out that way, it's quite straightforward. And sometimes we look for guys that may have a duplex configuration, 2 copies of R. That's another beauty about autotetraploids. You can get duplexes out of simplexes. Simplex means the QTL was present in 1 dose, and a duplex now has 2 doses. It's because of double reduction that we can get that product. It's the case when sister chromatids migrate to the same gamete, which then fertilizes another gamete, right? You can only do that with tetraploids, not with diploids. You'll never get a homozygous derivative from a heterozygous parent, right? But again, we can do it in autotetraploids. Okay, so this is an example of that individual that had the 2 QTLs for linkage group 3 and 5 for black spot. This is the QTL coming from migro. This is the QTL coming from bright eyes. And the way you interpret this graph is that the first 4 homologs come from one parent, the other 4 from the other parent in both cases. And the blue bars show you that that particular segment of that homologue is present in that line. So for any given section, there should be 4 bars that you can count. 1, 2, 3, 4, 1, 2, 3, 4, etc. And now you know, yeah, you've inherited the piece that carries the QTL in that particular individual. So we need to do that to make sure that we've got what we said we thought we got. Okay, this is the example for Rosebud disease. We found a the QTL on chromosome 5 again, but it's from homolog 4, which is different from the homolog that contributed black spot that was homolog 3. So now we know it's on 5, it's a different region, but it's also a different homolog. Good information to have as you're planning what to do with that material. And here we see that we've also detected individuals that are duplex from this particular combination. And that's what a duplex look like if you're going to look at its haplotype. It's got 2 copies of a particular segment and then 3, 4. And then for the rest of the chromosomes, it's basically single copy segments of the parental lines that contributed to the line. Why are we so excited about duplexes? Is that if you cross a duplex with a susceptible, about 85% of the progeny, 80 to 85% of the progeny are expected to inherit that resistance locus at least once. So duplexes are really good QTL donors. Most of your progeny are going to be inheriting at least one copy of the QTL. If you use a heterozygous diploid donor, only 50% of the progeny are expected to receive the QTL at least once, right? So again, polyploids can be useful in guaranteeing transference of a QTL through crossing, for example. So finally, this is what we do if we have all these QTL for various diseases. I got Cercospora, black spot, and rose-rosette disease. I know how many individuals have 4 QTL, 3 QTL, 2 QTL, 1 QTL, and the different configurations. QTL is present where 1 means QTL present, 2 means the QTL is in 2 doses, 0 means the QTL is absent. And then next to it, you have the number of individuals that had the particular configuration. And we have about 363 individuals here. If we do a disease index calculation and then plot it against number of QTL, we see that nice correspondence. The more QTL you have, the more disease resistance you see. Therefore, we want to start looking at individuals that have multiple QTL and start retesting them and seeing what else we have to do to ingress them into the breeding program and use that as increases in disease resistance over time. That's another way to validate that the QTL appear to be useful, okay? And this is one of those individuals with 4 QTL. And you should be acquainted with the way we do this now. This is 1 chromosome, 1 trait, another chromosome, another trait, and so forth. And that's where the QTL are. So we calculated what is the probability that we would have an individual with 4 QTL, if those are the probabilities, 6%. And we got exactly 10 out of 161, which means the transmission of this QTL is quite normal in this population too, which is another good thing to have. You want favorable alleles to transmit. We did a comparable experiment with diploids. And again, we had Shushiropi. It's a diploid variety. And we crossed them with 3 different breeding lines. I said we, it was actually crossed before I came here. But we took this interrelated population and pushed them through the same analysis for Cercospora, black spot, and rosette disease. And again, we identified materials from 0 to 4 QTL. These are the ones with all QTL. Of course, we're going to start now reevaluating. This will be the most resistant material. And again, there's a good relationship between QTL number and disease index for those 3 diseases. So now we know at least we have a subset of materials that we can say these are going to be the most resistant material or carrying disease factors for resistance. Because in this case, we're talking about QTLs. All of them provide partial level of resistance, not immunity. So individuals that have them have a greater probability of having resistance over those that don't have them. But it's not an absolute situation. As I said before, there are some major genes for resistance for black spot, and I have a graduate student, Joey Fu, who's been working with the one that has been cloned. So she's looked for used markers developed by others, and we developed some markers ourselves. And she started screening some of the material that we knew should carry RDR1, but we didn't know which ones had it. And she's identified about 44 breeding lines that have it. She's also screening varieties, and we have about 30 that appear to have this gene too. And we want to use those materials to, again, assess how useful RDR1 is in our environment by testing it against black spot or looking at historical data with that respect, and then trying to use that information to move and start pyramiding these resistances with RDR4 that we know it's in a different population, and then RDR3 with a new population that we're analyzing now too, and maybe bringing multiple disease resistances measured gene resistances is a strategy on top of these minor resistances or QTLs. All right, so that's all about diseases. And I had to show you something of ornamental value since we're working with roses. And this is an interesting trait that a graduate student, Amrit Gill, is working on. And this is what we call the flower color transition trait. And this is such a situation when flowers initially open, they're either yellow, light yellow, or white, but then as they age, turn dark pink. So if you look at that bush at any given point in time, you will see that it has flowers of multiple colors: yellow, light pink, and dark pink. So this is actually one bush, not multiple bushes together. So it's a very striking phenotype. This trait was segregating in some of the populations we were studying for disease resistance. So Amrit decided to start measuring that trait to try to understand the genetic basis of this particular color transition phenotype. So she took visual scoring. She looked at a rose and said, this one is transitioning or not transitioning. It's transitioning from yellow to dark pink, or from yellow to light pink, or not at all. And then she scored them 1, 2, 3 that way. And then she also measured color with a colorimeter, took the L, A, and C color space values, calculated hue and chroma, and then we used a flower color difference statistics, delta E. And delta E is used in industry to compare two objects to see if they have the same color or not. If they have the same color, delta E is very small. They have different color, the delta is very large. So a transitioning rose will have very large delta E's when you contrast the color between various flowers. A rose that doesn't transition has uniform color pigmentation, has very small delta E's. So that's an indirect way to look at flower transitioning. So you'll see A, L, B, H, C, and delta E in the QTL plot here. So we've got QTL for transitioning scores and delta E calculations in many places. The circles mark places where there's a lot of coincident QTL. And in the boxes, we have the location of genes that we know are involved in anthocyanin biosynthesis. And there's very good coincidence between our QTL and those genes. And we're excited about places where our QTLs overlap with regulators of anthocyanin synthesis itself. So Amrit's going to look at the gene expression patterns in transitioning types and also compositional changes to try to connect that with the QTL that we see here. Because QTL give you genome locations, but they don't tell you much about mechanisms, which is eventually what we want to understand better here. Again, this is just a list of all the traits that we are interested in or we've already measured, but we haven't analyzed to the degree we've analyzed some of the other data. We are developing new populations looking for other sources of resistance to rose-rosette disease. We're interested in flower productivity traits, flower intensity during the summer, architecture, and of course, other flower color patterns. And roses have an incredible diversity of that as well. So we've got quite a bit of data for plant architecture. We need to finish that up. We've started developing new populations for flower color patterns, and the same is true for rose-rosette disease. And we do have flower productivity data that we'll be analyzing soon too. All right, so that's all about QTL mapping. And one of the things that we've made a big push through the Tools for Polyploids project is to move in the domain of biocomputation, where we've got a couple of very talented postdocs, Chris Taniguchi and Jiken Lau. Jiken used to be a PhD student in the program who is very computer and script writing adept. So it was a natural evolution for him to devote his, I guess, skills to biocomputational activities related to the program. Chris comes from University of Sao Paulo, a very talented programmer and computer scientist with a genetics background and upbringing. So it's just a perfect fit for this type of project. We're working in collaboration with Marcelo Molinari and Xiaobang Zhen from NC State. And one of the outputs of that activity and collaboration in a very quick turnaround was a new software package that is called View Poly. View Poly is one of the visualization software that I showed you at the beginning of my presentation. It takes output from mapping and QTL analysis software, combines it with genome reference data, and provides a way to mine your QTL and mapping results and dig all the way down to a genome region and perhaps candidate genes. Something that we did not really have for autopolyploids or for diploids for that matter. And again, depending on the type of data you feed it, you can look at diploids as well as tetraploid data. It's an amazing tool and we're really proud of it. And it's available through GitHub and CRAN if you want to take a look at it. The only restriction is that the output has to be from this particular software with respect to mapping and QTL analysis. I'm sure Chris has got ideas on how to make that broader, but this is what we have so far. Another piece of software that Chris and Jiken have been working on, and again, this is a collaboration with the Statistical Genetics Lab at the University of São Paulo, where Chris comes from, is to develop an integrated computational workflow to go from sequencing data to basically dosage calling and then linkage maps. And again, this workflow is nice because it gives you options of SNP calling software to use, as well as dosage calling software to use, or combinations thereof. And then we use linkage mapping to evaluate the quality of the data set for mapping purposes and downstream uses. This makes life a lot easier if you're going to go from sequencing data to mapping in one run, if you will, and to be able to gauge the quality of the data sets that you have. Still being developed. Chris wants to call it reads to map, but again, as soon as that is ready, it'll get, I guess, unveiled when it comes. Still working to try to add MapPoly and PolyMapR, which are the tetraploid mapping software to do the mapping component on this particular pipeline. All right, as far as tool development, as I said before, one of the things that we gained with respect to genotyping capabilities is to be able to do GBS in tetraploid roses. Tessa Hockhouse is our PhD student that's working together with Chris and Jiken to make this happen. Chris and Jiken handle the computational stuff quite well, and Tessa has been able to take the wet lab component, which is also quite important. Oftentimes people complain about GBS. Oh, I don't know what to do with this GBS data, doesn't make sense, can't map anything. I bet you that goes back to library construction. And Trish Klein and her lab have to be the absolute perfect library construction people, because they were able to construct libraries in one go and develop top quality data for 3 mapping populations in roses. So our average read length was targeted to be 50x based on the literature, and we got almost right on target. And when we did the UpDog Oracle analysis, showed that 50x or greater depth of read would give us very accurate SNP calling and genotype dosage calling in an autotetraploid. So in autotetraploids, we have 3 heterozygous classes and then 2 homozygous classes. Homozygous classes are very easy to call. The difficult thing are those heterozygous classes, and we find that we can do that with this quality run. 50x or better is what you need to shoot for an autotetraploid. And I think we're there. And this is the result of that preliminary run. We went all the way from sequencing to a linkage map of about 1,800 loci. We could have more, but decided to go with the best that we had. About 1,000 centimorgans, 2 markers every centimorgan, all 7 chromosome maps. This is a very complex figure because it shows you the map, but also the phase haplotypes of the parents in that particular population. So complete phasing of parents, complete phasing of progeny for this particular map. So we can tag and trace every single locus in that population. Amazing results. I was surprised. I thought we were going to sit on the data for a while. Next thing I know, they've got a map. I was very surprised. Okay, so to finalize, I guess this is the approach that we're taking. Again, as I said before, mapping genes, trying to understand the genetic basis of how those traits, mapping traits, trying to understand the genetic basis of the traits, and then apply those in sort of plant breeding context to make things better, and eventually move to genomic prediction. Okay? And those are the traits that we're targeting at this point. Again, don't forget to visit our website, and we'd like to thank the funders, USDA SCRI for Tools for Polyploid project. And thank you to a lot of people, some that came before me and come after me, that have contributed to all of the things that I've shared with you today. Believe it or not, it also gets cold in Texas. All right. Thank you.
Thank you, Dr. Riera-Lizarazu. We're going to take questions now from the crowd. So if you have a question, please raise your hand.
I'm in between you and the bar. That's a problem, isn't it? Yeah.
We got one right up here.
Thanks. Oscar, did I see under your color, I was trying to look at your transcription factors. Did I see high 5 and SPL9, which?
High 5 is there, and MIB10. And but none of the SPL9s are really being picked up. SPL9 is not being picked up, MIB10 is, and high 5 is. So, yeah. And as you know, we also realized through all the work that AMRIT has done in collaboration with the Klein lab or labs, is that the transitioning phenotype is UV sensitive. If those flowers are not exposed to UVA or UVB, they remain yellow. But as soon as they're exposed to UVA and UVB, they start turning pink. So the whole transition in phenotype is UVA and UVB mediated as well. Yeah. Not very well.
My question is actually related to the transition types.
Mhm.
What if you take those plants and plant them in several places in the US? Would you still see the same type of pattern, or do you expect to see variation because of the different types of light that they'll have in different places?
I think they'll behave very similarly, but they're definitely depend on the amount of UV that they receive. It's a requirement. So if there are differences in the level of UV, like if you're going to plant them here versus you plant them in La Paz in Bolivia, for example, they'll get really red in La Paz for sure, maybe less so here. So maybe the phenotype is going to go so quick that we just don't even notice that it's transitioning. Because based on AMRIT's research, that transition takes place within 24 hours, more or less. So it's a quick response. It's yellow today, but it's red tomorrow.
We have three question comments from the chat. Kind of quick. First one, is it possible for you to show or share your scoring chart for the black spot in the field? They're also working with black spot and they use a 0 to 6 rating. They think they want to compare. Is that
Sure,
something you can share?
that should be possible. Yeah.
Okay.
They
And
just need to write an email to me or to Dave, and we'll share
okay,
it.
send you an email.
Yeah.
Second question, is View Poly available? I believe you answered that. Is that available for external use?
Yeah, it's available publicly through CRAN and
And
through, GitHub.
the third resource question is, do you have a paper on the QTL and disease level relationship that you can read up on? That
I'm
this person
sorry,
can read up on?
the which one?
QTL and disease level relationships that you spoke about. Are there papers you could direct them
we
to?
are getting the rose rosette disease papers finished up, and then we're going to have the black spot, and then we're going to have the sarcospora. Yeah, they're coming up.
Do we have any more questions? All right, join me in a round of applause then.
Yes, thank you.
So that was our last speaker for, that was our last speaker for the night. So I'm going to turn it over to Catherine, our fearless leader, for the closing remarks.
Very happy to be back in person this year. Thank you all for staying with us once again. We are glad to be back in person this year, and I would like to ask all the committee organizing team to please stand up for recognition. They have dedicated a lot of time and effort to make this symposium a success. Fabian? Okay. Can we please just clap for the organizing team? Yeah, so they were really, I don't call them like committee members because it was great to work with them in a team because every single person did their part of what they needed to do to make the symposium a success. So next, I also want to thank our support staff from Soil and Crop Sciences for their effort in getting some of the logistics and for a smooth ride today. Beth and the business folks for helping to get the money to fund from our sponsors to fund the symposium. I also especially want to thank the keynote speakers who flew all the way from their various locations to be here with us. We're really grateful and we enjoyed all your talks today, and we hope to have more conversations with you tomorrow at noon. We also want to thank our Cotiva travel scholars for being with us and sharing part of our research, their research and interacting with us, as well as the Texas A&M oral speakers, including our only undergrad winner. And we encourage you to continue the good work with your research in grad school. We also want to thank every single volunteer who has helped us today, including all the judges for taking out time to be here early in the morning by 7 to judge the posters, as well as every McGill and Rachel who have been here the whole day and parts of yesterday, moving around to do a lot of logistics. Thank you all very much for your support and your contribution to the symposium. And of course, without money, it is almost impossible to run this kind of events. So we want to thank all our private companies and Texas A&M departments who committed funds to support our symposium this year. We thank Cotiva, NUPI, Advanta, Texas AgriLife, Texas Corn, Peanut Producers, Soil and Crop Sciences, Horticultural Sciences, Institutes for Genome Sciences and Society, and the Department of Biology for funding this year's event. Thank you so much for your sponsorship. And of course, at 6 o'clock, we have the evening social, which will be at Moe's Irish Pub. It will be outside and the temperature has dropped, but there will be space heaters. So it will be warm. It will not be very cold. So please be there to interact and network. We'll also be announcing the winners of our poster competition there. So the social will run from 6 to 9. So you can take a break and meet us there. And of course, tomorrow we have the plant breeding and genetics circle from 12 to 1. So we will also have all our keynote speakers alongside with Jason, who with Jason tomorrow for panel discussion. So feel free to join us in HEAP room 440. We'll have a virtual option. So if you're joining us virtually, you can still connect and join the conversation. Thank you all once again. And indeed, we've come to the end of this year's symposium. Thank you all very much.